Good morning, good afternoon, and good evening to more than 200 people across the world who are watching this webinar and also who are live. I am Jennifer Smilowitz. I'm the director of the IMGC, the International Milk Genomics Consortium, and I'm excited to bring to you our annual spring Roll Up One Sleeves or ROS workshop. This workshop is hosted live, but you can also view it as a recording, and it'll be available to you on July 1st on our website, which is shown on the bottom left-hand side of the screen. Now, many of you know our origin story, but for those who do not, we are a scientific society created in 2004 by Professor Bruce German at UC Davis and colleagues from all over the world to make one discovery, and that is to sequence the lactation genome, which was accomplished in 2009. Now, today, the IMGC is made up of hundreds of scientists and innovators worldwide from academic and industry sectors, and we all work on the common goal of advancing lactation and milk science to improve human health. Now, we advance the field leveraging 4 platforms. The first is our flagship annual conference hosted every fall, and it features cutting-edge scientific research and accelerates scientific innovation. Our symposium attracts a diversity of disciplines and fields. It fosters collaboration, and it increases the impact of publications in milk science. Our second platform is our awards program, and this highlights scientists across career timelines, from students to legacy pioneers in the field. This platform encourages ongoing engagement with our base. Our third platform, as you may know about, is our bi-monthly publication called Splash Milk Science Update. And we translate the latest scientific discoveries in milk science from primary scientific peer-reviewed literature, and it's written by scientific writers. These are professional science writers. We have published more than 500 articles on milk science that are searchable on our website. And finally, our fourth platform applies to today's event. It's the annual Roll Up One Sleeves or ROS workshop, and it's a practical 3-hour focused webinar that addresses translational issues in milk science. And the goal is that you come away with a better understanding of bringing discoveries to practice, whether concepts or products for human health. Now, this year's topic is quite timely as the demand to preserve or safeguard milk bioactivity has become front and center in today's clinical care practice and also in mainstream media for its role in supporting both maternal and infant health and also the broader public across the lifespan. Now, to help us select next year's ROS workshop topic, please scan the QR code on the left-hand side of your screen and just take 5 minutes to fill out our exit survey. Okay, so this year's ROS workshop is hosted as a webinar with back-to-back presentations from 6 world experts in chemistry, nutrition, dietetics, food science, and engineering. And they will each share their experience, their expertise, and insights into the challenges of safeguarding milk bioactivity, along with their innovative solutions that they propose to overcome these barriers. After the last presentation, we'll take a 5-minute break, and then the panelists will rejoin in an open panel discussion for about 1 hour. Please submit your questions or comments in the Zoom Q&A function anytime during today's webinar. They will be addressed during the open panel discussion at the end of the webinar, at the end of the presentations. Now, without further ado, I am honored to introduce to you our moderator of today's webinar. That's Professor and Chair Alan Kelly from the University of College Cork in Ireland. Take it away, Alan.
Thanks very much, Jennifer, and I'm delighted to be here and to moderate what I'm positive is going to be an absolutely fascinating session today. So I join Jennifer in welcoming everybody from time zones all over the world. It's 4 o'clock in the afternoon in Cork in Ireland, where I'm joining from, and I'm a dairy scientist who's worked my entire career in the biochemistry of milk, both human and bovine milk, and on the processing of both. And I know that as Jennifer has said, this is such a key topic today uh, in every going from the dairy industry to the preservation of milk for preterm infants. Everybody is focused on how do we preserve the biological functionality. So there's a really topical area for us today. We've got a fantastic lineup of, as Jennifer said, world experts on the topic, and I'm really looking forward to the presentation. So, to kick things off, I'm going to introduce Professor Casper Hettinga to give our first presentation. And Casper is a professor in dairy processing and functionality at Wageningen University and Research in the Netherlands, where he leads the food quality and design department. And he's going to kick off by talking on thermal and non-thermal processes and their impact on milk proteins and their bioactivity. So, Casper, welcome, and I'm happy to hand the floor over to you if you'd like to start sharing your slides. And again, to reiterate to everybody, please put questions as you think of them into the Q&A, and we'll have plenty of time for a very, I'm sure, enlightening discussion afterwards.
Thanks a lot, Ellen, for that kind introduction. As Ellen said, I'm going to talk about the impact of thermal and non-thermal processes on milk proteins and their bioactivity. And where usually in presentations we talk about all the cool things we do and the nice findings we have, today I will talk about three problems. And that's not only from the perspective of the problem as such, but also how my thinking has evolved on these topics, because I've made assumptions in the past as a scientist that I don't look at in the same way now. And by sharing these experiences, I also hope you learn something not only about the topic, but also more in general about how to deal with doing research in this area. So as a disclosure, I'm a member of the scientific advisory board of three different companies. And today I will be presenting some outcomes from different research projects that have been funded through different external funding bodies. So as an introduction, we talk about bioactive proteins here, and bioactive proteins is a very broad name, a very broad category of components. And what we are interested in is actually a whole range of different components. So they may be relatively high abundant proteins, but especially also their peptides that are formed upon digestion that have specific bioactivities. But milk contains a huge number of low abundant bioactive proteins. Over the years, thousands of these proteins have been described, all with different functions, and they range from relatively more abundant and more studied ones like lactoferrin immunoglobulins to also very low abundant ones that have immune functionality, antimicrobial activity, but also stimulating development of the gastrointestinal tract and even for brain development. So there's a lot of different functions that are associated with these bioactive proteins. And if we then look at the processing of milk, these proteins change, first of all, structurally and chemically, but these structural and chemical changes that occur to these milk proteins also have an impact on the functionality, as I will explain a bit later in my presentation. So if we look at whey proteins, they may unfold and aggregate with themselves, but also they often aggregate on casein micelles, which is changing the structure of these casein micelles, but also how these casein micelles, as well as the whey proteins on them, digest upon consumption. And as dairy products usually contain lactose as well, there may be chemical reactions between the proteins and the lactose, leading to glycation, which is the binding of the sugar to the protein, which can then also be further chemically converted into all kind of chemical modifications of different amino acids. And in turn, this can also lead to aggregation. So different proteins may be bound to each other through these glycation reactions. And in dairy processing, all these things happen at the same time, but not to the same extent. So, for example, denaturation aggregation happens more in liquid dairy systems. Glycation happens to a larger extent in drying of dairy products or storage of dried products. So there's always a different ratio of these different components that is present. So the first challenge that I want to discuss today is if we know that these changes are happening from unfolding, aggregation, and glycation, and now heating and reducing sugars such as lactose are involved in that, the question is, what is that doing specifically to the bioactive proteins? Because on the previous slide, I showed whey protein in general and the caseins. But as I said, milk contains a huge number of different proteins. And generally speaking, protein aggregation reduces biological activity, also has effects on the technofunctional properties. I will not be talking about that more today. And generally also reduces digestibility. On the other hand, protein unfolding without too extensive aggregation may enhance digestion. So it really depends on which reaction happen and in which ratio these different reactions happen. What in the end the consequences are for the person that is going to consume these dairy products. So what we often want to understand is the relation on one hand between these changes that are occurring to the proteins to the functionality, that we really understand on a chemical level what is changing and what the consequences of these changes are. And when I started my research in this area around almost 20 years ago, I started this really from a protein chemistry point of view. So that is shown on the slide on the left. So by looking in detail at the amino acid sequences, the protein modifications that were happening, I was trying to solve these problems because I assumed that as long as we are able to very detailed analyze what is happening to proteins, which proteins aggregate with each other, we are able to solve this question of what is happening during processing. The only challenge is that not all changes that happen in dairy are easily shown by protein biochemistry. For example, changes in casein micelle structure or interactions between whey proteins and caseins are not that easy to detect, even though they do have large impacts on digestion. So over the years, I moved from a more protein chemistry point of view, which is a very analytical point of measuring what is going on, to linking that to digestion and different biofunctionalities. So by looking with different types of in vitro digestion systems and in vitro immunology systems or other types of functional assays, we try to really link these different things to each other. So in the end, to understand how processing impacts the functionality, it's good to have good abilities in all these areas. But in the end, if we really want to understand what is happening, we should be able to link these different types of analysis to each other. And that generally also requires collaboration with experts from different areas as well, because I don't believe you can really be an expert in all these areas simultaneously. Then the second challenge is related to establishing what the effect of thermal processing is. And we know, as I already mentioned before, that aggregation reactions, glycation reactions can have all kind of negative consequences. So the question is, how can we minimize such loss in bioactivity? And this is true for both bovine and human milk, although the way that these products are processed is, of course, very different from each other. And that actually leads to another misconception I had when I started this type of research, because my underlying assumption was that heating just damages protein. So we need to find ways to make milk safe without applying heat. but there's a huge impact on what temperature we process milk at. So if we look at milk processing, there are legal limits on the lower end to assure safety, but most dairy products are heated at a higher temperature. And the exact temperature at which you heat does make a big impact. And if we look, for example, at immune proteins like lactoferrin, immunoglobulins, we see that low pasteurization at the lowest end of what is legally allowed actually works relatively well. We can retain these proteins to a large extent in their native state. So even though, generally speaking, heating leads to the destruction of bioactive proteins, it, one of my insights is that it's more nuanced, with especially low temperature heating not being as detrimental to the quality of milk. And there is really room to improve. And this is from the bovine milk perspective. From the human milk perspective, there is another thing that plays a role, because if we look at human milk banks, generally Holder pasteurization is applied, which means half an hour at 65 degrees, and we are heating bottles of milk. And the heating up time of Holder pasteurization is about as long as the pasteurization itself. So here there are different heating methods, from HCST, which is generally applied in industry, flash heating, heating in a water bath at different temperatures, to hold the pasteurization at the bottom, where you need about half an hour to heat up before you apply your real pasteurization for half an hour at 65 degrees. So what we wanted to study here is look at these different processes which have different heating up times, as you can see in the middle column in the table on the left. And we also had Holder pasteurization, where we actually did the heating up in HTST equipment. So there you have an extremely fast heating up. And what the result showed is that does make an impact on protein retention, but especially HCST, so both a high heating up temperature as well as a very short heating time led to a much lower destruction of different bioactive proteins than higher or longer heating up times or Holder pasteurization. So temperature profile in the end is key to understanding what damage you are doing. And there are really big steps to be made with better thermal processing in human milk banks to lower the extent of damage occurring due to the heating. But next to thermal processes, there's also a lot of research going on in the area of non-thermal processes. And there are a lot of different processes that can be applied here. So UVC, ultrasonication, microfiltration, pulse electric fields, high pressure processing. So there's a whole range of different non-thermal processes. And the question is, what is their effect on milk proteins as well as the bioactivity of these milk proteins? And also in this area, my thinking has evolved over time, because initially I assumed if we do not heat milk when we apply such a non-thermal process, we are also not causing any damage to milk. But that's actually not the case, because what we saw, if we look with proteomics, is that different non-thermal treatments actually also already start causing changes in the proteome of human milk. And also, if we look at functionality, so for example, the antibacterial activity or the blood clotting ability, we do see large differences among the different non-thermal processes. So it's not that all non-thermal processes are equal to each other. And especially when we look at functionality, and I only show two examples here, but we have actually done several others as well, we see big differences in their impact on functionalities. And sometimes, for example, with ultrasonication, we saw damage to the proteins and protein functionality, especially, that was similar to Holder pasteurization. So even though you do not apply a traditional heating process, the damage done to functionality is the same. And this is something that also relates back to what I said at the start of my lecture, that we need complementary methods to understand what is happening during processing. Because if you actually look with traditional proteomics based on LC-MS, some of these methods do not show a decrease in certain proteins, so you would assume based on that that no damage is occurring, whereas functional assays do show changes occurring. So we really need complementary analytical approaches to really understand well what is happening, and especially for non-thermal technologies, we may need further or different types of tests than for traditional heating to be able to understand the damage that is caused by the process. So all these non-thermal technologies have on one hand certain advantages, especially having a lower impact on bioactive proteins, but also a lower impact on all kind of other uh, properties. But most of the techniques also have disadvantages, which can be a high energy use, or they can lead to other negative effects on the quality or the techno-functionality or the flavor of dairy products. So you really need to weigh these pros and cons of these different methods to be able to select the optimal method. And that may actually differ between human and bovine milk. And then I also have a solution 4, which is a solution for challenge 2 and 3 combined, because I already mentioned for challenge 2 that a solution may be to go to as low as possible treatment temperature, even though that may not inactivate all the microbes. At least it does inactivate the pathogens, but other bacteria may survive. But by combining such a very low temperature treatment with non-thermal processing, or by combining different non-thermal processes sequentially, we may actually achieve a higher microbial inactivation than with traditional heat processing, while we have a reduced decline in our bioactive proteins. So in this way, by combining different processing steps, we may be able to try and achieve the best of both worlds. So the conclusions of this first lecture are that, in my view, we need interdisciplinary approach to be able to link these different types of analysis to better understand the mechanism. So we do need the protein biochemistry, we need to study digestion, we need to study bio functionality in a coherent way so that we really get a better grip on how the structure and the chemistry of proteins links to their functionality. Second, reduce protein damage in general, no matter whether we talk about thermal or non-thermal methods, translate to actual retention of biological activity like bacteriostatic properties, but also immune responses of immune cells. And non-thermal technologies reduce heat-induced damage to proteins to a different extent. So it's not true that all non-thermal technologies protect all proteins equally. There are large variations amongst the different techniques. So then my final slide is about what the future may bring. So when we look at challenge 1 and the bioactivity, the question is, do we ever know enough? I mean, there are so many different functionalities, there are so many different processes. So I believe that we have to be clear on which functionalities are most important, also on a global level, to select the most relevant ones. So by meeting and discussing as scientists globally, we should select those functionalities where we really would like to make a difference. And then for challenge 2, I believe that optimizing heating process, potentially also combining them with non-thermal processing, can lead to better retention of bioactive proteins. But on the side of the non-thermal proteins, we really need to have a better understanding at a molecular level what is happening, because these non-thermal processes are not are still changing the proteins. And if we are able to do that, we should also be able to relate this to digestibility. I talk more about functionality than digestibility today, but in the end, digestibility plays an important role as well because dairy products go through digestion in most cases before they deliver certain functionalities. So by being able to tailor digestibility, we may be able to achieve improved functionality of dairy products. And with that, I would like to thank you all for being here, and I give the word back to our chair.
Thank you very much, Casper, and perfectly on time. So thank you for that, and hopefully all speakers will follow your good example. That was a great start. I think it really set the scene, and it raised a number of themes and issues I'm sure we're going to come back to. And I see that we already have questions coming in on the Q&A, which is great. So I'd remind everybody to please feel free to enter your questions, and we look forward to answering them later. But for now, without further ado, I'm delighted to introduce our next speaker, who's Dr. Dave Dallas, who is Associate Professor of Nutrition at Oregon State University, where he's also the endowed director of the Moore Family Center for Nutrition and Preventative Health. And Dave has done a huge amount of work in human milk, but in particular in parallel in bovine milk. And today is going to talk to us about improving quality of donor human milk by alternative processing to enhance health outcomes for preterm infants. So a critical topic, and I think a really relevant one for today. And we're going to be, as we'll see, switching between bovine and human and comparing, because I think a really important theme for us today is what can we learn from the two different communities and two different approaches. Okay, so Dave, are you good to go?
Yes, I'm good to go. Just 1 second here. Okay, share. Okay. Well, hi everyone. Thank you so much for joining us today. So yes, I'm going to talk about human milk specifically. I do work on bovine milk as well, but this talk is really focused on human milk and some of the alternative processing methods that Casper was mentioning earlier. And I don't have any conflicts to declare for this presentation. And so I focus a lot on preterm infants. In the US, at least 10% of infants are born preterm, which is less than 37 weeks of gestation. And these babies are at increased risk for a variety of issues like poor growth, poor neurological development, infections. And part of this is because they have a reduced digestive system capacity as well as a immature immune system functioning. And some of the focus that I'm going to talk about here is really trying to help with this reduced digestive capacity through optimal treatment of human milk. And so for preterm babies, it is recommended to consume for them to be fed parents' own milk as opposed to formula because it helps reduce the risk for the various states of disease that are associated with prematurity, like necrotizing enterocolitis, sepsis, and other neurodevelopmental delays. And this is in part because of the variety of bioactive compounds that are present in human milk. Casper mentioned some of these. There's proteins that have antimicrobial activities, some that help assisting in digestion, some that assist in absorption of nutrients, some that help guide the development of the infant's gastrointestinal tract, and some that are immunomodulatory. And so one of the issues, however, is that preterm parents who deliver preterm infants, very often can't produce enough human milk to fully feed their baby. So I think it's over 60% of parents are unable to provide enough human milk to feed their infants. And so human milk banks fill this gap in milk availability. And so in the US, at least, most preterm infants in neonatal care units are fed at least some donor milk. And that amount is increasing over time. But as Casper was mentioning, there's a challenge in that donor human milk has to be processed to ensure microbial safety because donor milk could be contaminated from a variety of sources, maternal skin, from the pumping devices, just handling processes in general. And so in order to ensure safety, donor milk banks have to do some sort of processing to reduce any sort of microbial load that might be present in the milk. And so some kinds of processing that are used include Holder pasteurization, which is 62.5 degrees for 30 minutes. That's done at nonprofit milk banks in like individual bottles. There's also vat pasteurization, which is the same thing, but just in a very large scale in a single vat. And that's done at some for-profit milk banks. And then there are two other techniques. One is called ultra high temperature short time sterilization, which is higher temp 142 for just 3 seconds, short time. And then retort processing, also high temp 121 degrees Celsius for 5 minutes and with some pressure, 20 psi of pressure. And both the UHT, ultra high temperature, and retort processing create a shelf-stable product that does not need to be frozen while in storage like the Holder pasteurization or vat pasteurization products. And that's because they create a greater log reduction of spores that allow it to be shelf stable. So we did this experiment comparing these different treatments, the ultra high temperature pasteurization, vat pasteurization, and retort processing compared with raw, and also we included with and without homogenization because some products are using those. So overall, we found that immunoglobulins are better preserved by this vat pasteurization, which is sort of equivalent to Holder pasteurization, compared with retort processing and ultra high temperature processing, which fully destroyed the immunoglobulin A. And similarly with IgG, we found that UHT and retort were much more damaging. With IgM, same story. With elastase, which is a protein digesting enzyme that's pretty abundant in human milk, and we think it's important for protein digestion in the infant. All the processes caused the degradation of this enzyme, all the thermal processes. Lactoferrin, we saw pretty high degradation with retort and UHT, and some with VAT PT, but a little bit better. And then from a proteomic perspective, this is a heat map just showing all the proteins, and then the different samples are sorted with hierarchical clustering. So you can see here that in the middle, raw is most similar to the VAT pasteurization. But you can see there are some differences in that protein profile, and that the retort processing and the UHT processing are very different. They cluster separately, and you can see a lot of red where the protein abundances are very different after those processes. But even though I showed Holder pasteurization was better than those higher temperature processes like Casper was referring to, even that does degrade some proteins that are really important. And one of them that I'm really interested in is bile salt stimulated lipase. And that is a key enzyme that's present in human milk that is, it's present in the milk. It's not really active in the milk, but then when it enters the infant and reaches the intestine, it interacts with the bile salts that the infant secretes and it is activated. And this enzyme is very helpful in digesting fat in the infant. But all the processes, including vat pasteurization, completely destroy this lipase. And so the loss of that enzyme could lead to the lower ability for infants to absorb fat. And as fat is the main caloric source in human milk, that can really decrease growth outcomes. And some previous studies have shown that feeding preterm infants with pasteurized parent's own milk reduces the absorption of lipids by 20% and reduces need to heal length gain compared with raw parent's own milk. And some other studies show similar things with parent's own milk compared to donor human milk. So we are interested in some alternative processing methods. So here I'm showing high pressure processing and ultraviolet light processing. So I'm going to talk about those. So the first solution here I'm going to talk about is high pressure processing. So this is a technique that basically puts the liquid container in a flexible container. It's placed in basically a water bath that is squeezed and compressed, and that compression translates through the milk and into the things in the milk, including bacteria, and it causes disruption of bacterial membranes. And this disruption causes them to die and prevents replication. And so this can be typically between like 100 megapascals to 800 megapascals, usually without heating, and it can range in time. And so we did this experiment to try to identify what would be optimal pressures and times for high pressure processing to ensure safety and retain components in human milk. So we looked at some different strains of bacteria that we thought would be relevant for human milk contamination. And we found that 500 megapascals for 9 minutes at 4 degrees C was sufficient to cause greater than 5 log reduction, which is the target we're looking for pasteurization, of all the potential pathogens that we tested, some gram positive, some gram negative. So that's great. Um, However, we found when we looked at spores, and this is not the vegetative form, but actually the spore form of each of these spore-forming bacteria, the reduction with high pressure processing is not very high. It's less than 1 log. So even though we can create a safe product, it will still need to be frozen and refrigerated. It's not shelf stable. So you need to have spore reduction to create shelf stability. Now, this is quite similar to Holder pasteurization, which similarly will provide 5 log reduction of vegetative bacteria, but it does not provide that for spores. Okay, so in terms of bioactive retention, we found that these various different doses of high pressure processing, even the one at the end here, 500 megapascals for 9 minutes, which we determined as the needed dose, they improved the preservation compared to Holder pasteurization, which is here, of biosalt simulated lipase, which is great for ensuring fat digestion can continue to occur. And it also improved preservation of other proteins like immunoglobulin A, lactoferrin, immunoglobulin G, elastase, that protease I was talking about, immunoglobulin M, and polymeric immunoglobulin receptor, which is an abundant immune protein in human milk. So we have recently conducted a study to see whether this makes a difference in babies. So we did this basically 1 day crossover study in preterm infants where we fed them either Holder pasteurized milk or high pressure processed milk, and these were from the same pool of donor human milk. And we fed that to them 1 day at a time. And then we measured the fat that remains in their stool by gas chromatography, fatty acid analysis. And we found that there was more residual fat in the stool of the babies after a Holder pasteurized feeding compared with a high pressure process feeding. So it demonstrated that the preservation of the biosalt stimulated lipase was actually beneficial for these infants. And I didn't include it here, but we've done some other studies and we've shown that actually you can see this intact active biosalt stimulated lipase even in the stool of these babies fed the high pressure processed milk. So the second potential solution is using ultraviolet C irradiation. So this is another technically non-thermal treatment, although UVC light may induce some amount of heat entering into the milk, but that can be controlled with temperature control systems. And so UVC energy is absorbed by DNA, and it causes modifications to the DNA that prevents the DNA from being replicated in bacteria. So it causes these uh, linkages between thymines that prevent them from being further replicated. So again, we tested a big range of different doses to try to find what would be the minimum optimal dose. We used UVC with a 252 nanometer wavelength, very tight wavelength. This is just a very simple system that's been used in literature before. You just stick a UVC bulb into a beaker and you provide mixing, continuous mixing on a stir plate. And that's important because milk is highly opaque. And so so the penetration depth of the UVC light is very low. So you need to have that milk circulating so that all of it can be equally exposed to the light. And here we found that we were able to achieve 5 log reductions of all the vegetative bacteria that we tested using a dose of 6,000 joules per liter. And then we were able to reach 5 log reductions of the spore-forming bacteria here in the brown and the dark blue at 12,000 joules per liter. And we also found that both of these treatments, the 6,000, which is for the vegetative bacteria, and the 12,000, which is for the spore formers, better preserved IgA, IgG, IgM, elastase, cathepsin D, and lactoferrin compared with Holder pasteurization. And similarly, we found that all of these doses of UVC light treatment enhance the preservation of bile salt stimulated lipase, as well as lysozyme, which is an important antimicrobial component of human milk. From a proteomics perspective, we also showed that Holder pasteurization is more different from raw milk than high pressure processed milk and UVC processed milk. So they have, arrays of, there's some proteins that are different in, across each of these groups, but overall more similarity between raw and HPP and UVC than HOP. We had some worry that there could be some lipid oxidation induced by the UVC processing, and that is a reasonable concern. But it's possible that the doses that we're using may not cause that. And so we used an assay for measuring malone dialdehyde, which is a byproduct of oxidation of unsaturated fatty acids. And at the doses that we use, we found no significant increases in that compared to the raw. And so that indicated that we did not have an issue with oxidation of those fatty acids. And we also looked at vitamin E in two forms, alpha tocopherol and gamma tocopherol. In this case, we didn't see any decrease in gamma tocopherol, but we did see some significant but not super high decreases in alpha tocopherol. And so that could be an issue. And we're thinking that we may need to supplement the milk with additional alpha tocopherol either before or after to ensure that we don't have a reduction in the presence of that in the human milk. We're also looking at other techniques for providing this UVC processing. So we're working with Ankit Patras's team at Tennessee State University, who has a more higher, larger scale system. It's called a serpentine Dean flow UVC system, where milk is passed through very thin channels with specific angles that will cause Dean flow vortices or mixing to ensure optimal mixing of the milk to deal with this opacity problem. And we have it. This is a scalable system that can easily meet donor milk bank needs. And so far, same kind of demonstration. We've demonstrated that, we found doses that will provide greater than 5 log reductions of the vegetative bacteria we're testing, and that those doses can better preserve a variety of different proteins of interest. Another approach that we're just getting into now is microwave processing. And this is kind of similar to what Casper was saying earlier. If you're providing high temperatures over a shorter amount of time, you can see better preservations of protein. And so we are using microwave as just basically a way to get temperatures up very quickly. And so we tested different times, 1, 2, and 3 minutes using 300 watts. And we found with 2 minutes, we could achieve the dose required. And this was able to better preserve a variety of different milk proteins compared with HOP. So overall, good progress here, but there's a lot more future research that's needed to measure the outcomes of these on more bioactives and also measure their clinical outcomes before we can really have adoption of this at a larger scale. And with that, I will acknowledge all the people who helped with the work I presented here, and I'll pass it off to Alan.
Great. Thanks, Dave, for a great presentation and again raising lots of interesting questions and topics I'm sure we'll come back to later. So that's great. And we'll move on to our third presentation. And while we were discussing the program for this, we thought it was really important to get lots of different perspectives, but a critical voice we wanted to have was the industry practitioner perspective. So I'm delighted to introduce Harold Schouten, who's a senior scientist in preservation technology at Royal Friesland Campina in the Netherlands. Um, and he is going to speak to us about challenges in applying non-thermal microbial reduction technologies in protecting heat-sensitive proteins in the dairy industry. So, Harold, I hope, and apologies if my dogs are barking in the background, but I hope you can
No worries.
hear. And Harold, if you'd like to share.
Yes, I will share my presentation. Thank you very much for the introduction, Alan. Yeah. Yes. Now it's there. Huh? Okay, thank you very much, and also thank you very much for the invitation to get the perspective from the industry. So, as Alan indicated, I work for Friesland Campina, which is a big dairy company, dairy cooperative, where we produce, of course, all kinds of dairy products. And part of our portfolio or part of our aim is also to try to develop products where heat-sensitive proteins and bioactive proteins are present. So I will share some of my insights on how we deal with all the research that has been done from a nutrition point of view, but mainly also from a technology point of view, and how can we implement that as Friesland Campina or as dairy industry. So disclosure, the only disclosure I have, of course, is that I'm an employee of Royal Friesland Campina. So let's continue. So, preservation technology specialist or expert or whatever, what does it mean? So I look within Friesland Campina from raw materials to end products, and how do we design our processes to meet the specifications? On one hand is the specifications for microbiology, for instance, shelf life for microbiology and powders. And on the other hand, we have all the other product specifications. That could be taste, that could be structure, but more importantly for this talk is the bioactive proteins. If we want them in the products, how would we do that? How would we design our processes? And especially we look, of course, to unit operations, maybe novel non-thermal technologies, but also how do you integrate that in the whole production line? Like I said, this is really a balancing act. We use a lot of heat, of course, to make sure that we make microbial safe products or with a certain shelf life, shelf stable products. And heat is there the most common used thing to do, and to make sure that bacteria that you want to be inactivated are inactivated. However, this contradicts really towards keeping heat sensitive bioactive proteins intact. So this is really a balancing act on how to do that and how to manage that. And it's indeed, we use different unit operations, but it's really how to integrate it. And I want to show some insights that we have on how to do that and how to cope with these things. Just as first, a simple example case. Say we want to make milk powder. And this is really an example case and simplified. But what we normally do, so as a company, we take in raw milk, and just to make sure that we do not that we inactivate pathogens and we do not spread pathogens further within the factories, we pasteurize it first. So 18 seconds, 75 degrees is a normal industrial way of putting the settings, meeting, say, the 15 seconds, 72 degrees, which is applied by law and just the pasteurization conditions. After that, normally we have a high heat treatment. I just have an example here, 5 seconds, 120, could be a little bit different. And then we evaporate the water and we dry the rest of the concentrated product towards a milk powder. And for instance, just to show, we have a spec that we have less than 1,000 colony forming units in the powder. So what happens actually towards the microbiology to make this happen? So for instance, we start with 100,000 CFU per ml, which is a bit high, but okay, let's challenge the case. We pasteurize, and then actually this pasteurization is done to inactivate the vegetative pathogens, but also you inactivate much more bacteria with this time-temperature combination, meaning that you have more or less 1,000 CFU per ml left. These 1,000 CFU per ml are more heat resistant than what the pasteurizer can inactivate. So with the high heat treatment that we normally do, you are left with, for instance, 10 CFU per ml. So these 10 are something like very heat resistant spores. If you then evaporate and dry, we concentrate actually also the microbiology, and then we are left with 100 CFU per gram powder, and we say, okay, this is fine. So this meets specifications. However, as also shown in the previous presentations, if we do this, we do not have any bioactive proteins left. So this is not the way to go to preserve bioactive proteins in, for instance, milk powder or ingredients. So the first thing we say, let's remove the high heat step. And if we then look to microbiology, we start with 100,000, still the same amount. The pasteurization lowers it to 1,000, and we actually do not meet our specifications, because then we end up with 10,000 CFU per gram in the powder. But we have heat-sensitive bioactive proteins left, and so this demonstrates where the need for non-thermal preservation technologies is. So we need to have something in place that removes the more heat-resistant bacteria that normally are removed by high heating steps. So I have some challenges to make this possible on a factory scale. As already shown in previous presentations, there are several technologies that we can use. So we presented heat. So I can use low pasteurization, as also presented in the previous presentations, and then I lose more or less 10 to 20% bioactive proteins, and that could be acceptable. But I also have other parts. So I can use bactifuge to remove spores. I can use pulsed electric field, high voltage electric pulses to reduce microbiology. I can also use high pressure technology, as also already presented, UVC technology as presented, and CMF, which is a microfiltration technique specially designed to remove microbiology within, especially also in dairy streams. So, but how do I know which one is the best to use in specific conditions? So, how I look to that, first of all, it needs to be fit for purpose, and I mean, I need to know what end product specifications do I want. Is it a powder with strict microbial specifications? So I need really high reduction of certain bacteria. Or do I don't have any loss of bioactive proteins allowed, or is some loss allowed? So that makes that's an argument to choose for one or the other. Secondly, I really need to know what my target organism is. So that my target organisms, the microbiology that comes from my raw material. It actually depends if I make for instance, a protein concentrate from a milk, or if I would make a protein concentrate for whey, so the byproduct of the cheese production. The bacterial species which are in milk or in whey are different. So I also have different target organisms, and depending on the target organism, you can also choose a different technology. So one technology will inactivate spores, and the other will not. For instance, if spores would be one of your target organisms. As being a company, also the business case is really important. So the technology should also be positive for the business case. So if it's really expensive, we will never do it. That's normal. And there are two parts of it coming from the technology. One is, of course, the investment cost, but the other one is also the running cost of the technology. And finally, it also needs to integrate into the production process. For instance, for human milk bank, just was showed that also high pressure could be an option. But high pressure, for instance, is really difficult to integrate in a high throughput, fully continuous factory with capacities ranging from 5,000 liters an hour up to 80,000 liters an hour of liquid flows. So also, therefore, it needs to fit the process. It needs to fit the capacities of the surrounding equipment. And also, it needs to fit, for instance, safety procedures and all these kind of things. Next is an important thing. So if I look to pasteurization, that we already use for more than 150 years, this is a really smart technology in that sense, because you can really guarantee food safety or a certain amount of reduction. So if you look to pasteurization conditions on low side, we exactly know what we need to make sure that we reduce a sufficient amount of pathogens in there. Or if you take high temperature processes, for instance, to make shelf stable milk, we know exactly what we need to inactivate sufficient spores to make sure that it's shelf stable. And how do we do that? We can really, in the process, that is integrated in the process, guarantee the time and the temperature that is needed for this inactivation. So the time is managed by the holding time, and the holding time is managed by the flow and the volume of the holder. And the temperature, you can measure directly in your product what the temperature is. And so you know exactly the time-temperature combination, and you can also record it and make sure that it's every time the same, and so guarantee food safety or microbial reduction. So if I project that on different kind of technologies, if I look to heat, I just discussed, we can reduce vegetative cells and spores, and we can really guarantee that process. Then I go to bacteriophage, so that is based on density and on gravity. Vegetative cell reduction is really is quite difficult. Spores reduction, that's the function of bacteriophagation, but we can never guarantee it. We don't know exactly what is happening, and we cannot record something that is indicative for the amount of reduction. For pulse electric field, could work for vegetative cells. For spores, I haven't seen the proof for that, but it's also difficult to guarantee. You can, of course, measure the amount of pulses and the voltage and the amount of energy that you put in, but the residence time within the pulse electric field cell, so compare it to the holding time in a heat process, is really short. So you are never sure that all the parts are treated. So I would be a bit cautious to really guarantee this process. HPP processes, as already presented, it's determined by pressure and time. We can inactivate vegetative cells. Spores is really difficult. You need combination technologies. But since it's a packed process, you can measure pressure and you can measure time very well. So you can also easily guarantee the process and the effect of it on microbiology. UVC technology, that is actually the light intensity versus the exposure time. It inactivates vegetative cells, and also shown in the previous presentation, with more energy, it can inactivate spores. But it's a little bit more difficult to guarantee, because you can measure the amount of UVC light energy coming from the UVC lights, but you cannot really measure it in your product. So that makes it difficult to guarantee. It's a bit of a indirect measurement that you do. And last but not least, ceramic microfiltration. So that's depending on pore size. The pore size should be small enough to make sure that the bacteria do not pass the filters. So you can reduce vegetative cells and spores. However, there is no way to guarantee that all the product in all the products there are no spores or vegetative cells anymore under the conditions that we use for milk. You're never sure that one of the cells, vegetative cells or spores, has passed the membrane. So challenge 3. This is really a typical topic for the industry. So new preservation technology, unknowns makes also unloved. Huh? This is difficult. So because also in my previous job, for instance, I worked also for the university on a contract research part and also on non-thermal preservation technologies. And then you think, okay, this is the solution to everything. If you do this, then we can reduce bacteria without heat, and everybody would love it. Now, welcome to the industry. That's not the case because there are a lot of questions and a lot of uncertainties taking that into account. So it needs a lot of convincing and explanation to make sure that people believe in it. And to do that, the solution is more like we need to build trust with knowledge. And the acceptation is really on different levels. So internally, if I want to say, okay, I want to, for instance, introduce UVC to make sure that we inactivate sufficient amount of bacteria in a non-thermal way, people are starting to asking all kinds of questions. What happens to the product? Do we still have the same specifications of the product? Do the proteins, are they getting denatured? Are there other components that are interrelated? So I need the science behind it to prove also internally, if I look to R&D and to our NPD teams, to make sure that we can do this. On another part, there are really legal challenges. So for different technologies, there are different challenges. Let's take the example of UVC again. So in Europe, it seems to be more or less acceptable. But as Friesland Campina, as a worldwide company, we produce for products for all over the world. For instance, for China or for Asia. And there the legislation could be very different. So we can produce here under European legislation, but we cannot sell it, for instance, in other types of countries. So this is a difficult puzzle to make. Another thing, so if we make, for instance, ingredients, so whey protein powders, whey protein concentrate powders, with that are going to be used by our clients, they like, of course, that bioactive proteins are present in it, but we also have to sell, okay, how do we make it? And they also should believe in the technology and the safety of the technology. And for last but not least, also our consumers. So if we say we are going to use a specific technology and that doesn't resonate at consumers, then we really have an issue because our products will not be sold. So also convincing consumers is also part of this. So if I look to future directions and scanning all these kind of for technologies for use in industrial applications for big dairy industry and big volumes, I see actually the following. Pasteurization, like I said, we did that for more than 150 years. It really keeps on being the basis for microbial food safety. An experience of more than 150 years cannot be replaced by saying, okay, we can reduce enough pathogens by a different technology. That really needs to be built up very slowly. So from my perspective, this will stay at least for 10 years and probably longer. However, to make products with intact bioactive proteins and to remove the thermal-resistant vegetative and vegetative cells and spores. The current best practice is to use ceramic microfiltration to remove that and to make sure that you have sufficient reduction of these bacteria on low temperature conditions. However, this process also has some disadvantages like high investment cost, high running cost, and also a waste stream. So your retentate is still there. So what I see, and as Friesland Campina, what we see is that UVC will probably be the most promising next practice, as well as from capex and from costs and running cost perspective, as from effectivity from a microbial reduction point of view. So it reduces also the more heat-resistant bacteria in dairy streams, as well as the vegetative cells as some spores. And next to that, as I can oversee, but I really need your help also for that, it doesn't affect the bioactive proteins present in milk and in whey products. So I'm really eager to see how this will evolve. So, and then finally, conclusions. To really make bioactive proteins within dairy ingredients and dairy products possible, non-thermal preservation technologies are key. So we really need that to do, to make all the science behind it, also from a nutritional perspective to make that happen. These non-thermal preservation technology we need. Like I told, knowledge is really key. We really need to know what happens with which technology, as also Casper indicated. So there are also some other reactions, other things happening that we don't fully understand yet with different technologies. You need to focus really on the target organisms. So you need to design it for what you really like, for what microbiology is in it, but also for the components that you are interested in. Some proteins might be more heat resistant than others. So you can also tune your process on that. And finally, we need to build trust for acceptance. So this it needs to be accepted from a legal point of view, but also people need to believe in it, as well as internally within a company as externally for clients and consumers. So hereby I want to thank you for your attention.
Thank you, Harold, and that was a very nice presentation, which really raised some very interesting practical questions about implementation and use in industry, and I'm sure will provoke a discussion later. And again, I'd encourage people, if you have questions, to put them into the Q&A, and we'll come to them in our discussion in a little while. So I'm happy to move on to our next presentation. Well, thanks again to Harold, and that's Professor Lilja Arne, who is professor at Swedish Agricultural University, SLU. Where she has recently moved from the University of Copenhagen, where she was professor of dairy process technology, and who has worked on the topic of non-thermal processing, as well as thermal for processing of milk and for preservation of biological activity. So we're delighted that Lilja will present now on enhancing milk bioactivity using non-thermal technologies. So Lilja, we can see your slides. Everything seems to be working well, so I'll hand over to you.
Thank you. Thank you, Alan, for the kind of presentation, and you have also enjoyed very much to listen to the presentations before mine. So I will bring a little bit a different perspective here, more from the process side. And I think everybody have seen eventually some of these products in the market. For example, if we do thermal pasteurization of some of these green juices, they turn more brown, not very attractive. And on the other side, no thermal technologies have shown that you can preserve this attractive color of raw materials. So there is, in fact, potential on these non-thermal technologies for acid foods, for example, and the juices, an example that have really made it to the market because of the requirements on pasteurization are not as high as with milk. But of course, then there is always there is a need for no thermal technologies in dairy processing. And I think the previous lectures have already shown some very concrete examples why this is important. And therefore, I still think also that many of these technologies have been primarily explored in relation to milk towards preservation. And also Harald explained, of course, is a very important aspect. But what I start to wonder a little bit, can they be pretreatments? Can they provide the possibility to develop health bioactive compounds that can have health benefits? So that is in that context that I will talk a little bit about both the use of pulse electric fields and UVC in this context. And I think pulsed electric fields, I think many of you have heard about. I know even that some that have worked with. So basically, you apply a high voltage for a very, very short time. And what basically this electric field then affects, for example, bilayer membranes. They try to orient, the phospholipids try to orient in the field, in the electric field, and then they end up breaking and you get electroperforation. And of course, this electroperforation depends very much on the type of membranes, depends on the size of the organism, and they can be reversible or irreversible. So, but of course, also associated with pulsed electric fields, you often have a thermal effect, especially if you apply this process for a longer time, you end up to having also a thermal effect. So in many cases, in fact, pulsed electric field is a treatment that combines both these effects of the electric field, but also the thermal effects associated. So my question then, we know, for example, that for milk, pasteurization or inactivation of microorganisms with PEF has not been very successful. I mean, it's difficult to get tangible advantages of such. But our idea was, what about regarding milk components? What do that mean? I mean, the pulse electric field affects membranes, as I'm saying. I just mentioned. What about the membranes of milk fat globules? Could that have an effect? And if does, what that would mean? We were also wondering about major component. Caseins are organized as a casein micelle, so it's a main structural element of the milk. What would pulse electric fields influence the caseins? And what about whey proteins? We have also a number of soluble whey proteins in the milk structure. Can PEF affect them in the way that can contribute for beneficial effects? So, I mean, if we look at the effects of pulse electric fields on dairy proteins, there is been quite a lot contradictory results from in literature. I mean, it's difficult to compare studies from different types of equipment because some equipment have more ohmic heating and they produce higher temperatures increase. So you can get all the type of results and conditions that have been used. So, and goes from everything, no effect on caseins until some effects on reducing size of caseins, and the same regards whey proteins, partial denaturation, no effects, and of course this depends very much how the process has been applied. So we did, oh, what happened? We did a study. So I just want to mention this is not our study. So it's a study done by Alexander Matis from ETH, where he applied pulsed electric fields and studied the effect on lactoferrin and the hemoglobin A. And he could see that up to a given amount of energy supplied, you can retain quite well the concentration of these bioactive molecules. But after that, for higher specific energy, as we can see here, marked with these lines, then you start to see a decline on the concentration of these compounds. So yes, it is possible to keep the bioactivity of these compounds, or these bioactive compounds, but depends also very much on the conditions that are applied. If we then look at the milk fat global membranes, and was our study, and in our study we hypothesized that, depending of how intensive is the pulse electric field treatment, you may have partial the electroporation of these milk fat global membranes, or you have quite a completely electroporation, so it comes to a point that milk fat global cannot keep its structure intact. So but we also know from milk that the size of these milk fat globals can be quite wide. They can change quite a large in size. And as this technology depends on the electroporation, depends also on the size of the globule, we decide to make a study where we separate by a two-step separation. We create milks with small milk fat globules and then milks with large fat globules. And then we subjected this reconstituted milk compared with raw milk, and then we submitted to pulsed electric fields at two different intensity. So 9 and 16, and then we try to keep the other conditions for the initial temperature and the final temperature quite similar. So, just to confirm, we could see from our results that, in fact, our process was successful. So the size of the small milk fat, or the milk with the small milk fat globules, this MS, was higher amount of these small molecules, and there was a shift to the left to small molecules, which is also confirmed here by this microscopy. So, but we could also see some differences here in the milk fat protein profiles between these the smaller and the larger ones. We could also see some differences, which was a little bit surprise in lipase activity, where the globules, the milk fat globules, the milk with larger milk fat globules, in fact, had a reduced activity of lipase. And of course, lipase can be associated to the surface of these globules, and probably that's also the reason for that. We look at with more detail also to the inactivation of PEF, of the milk proteins, and we could see in these conditions used very limited degradation of beta-lactoglobulin or alpha-lactoamine, and which was kind of expected because was not a very intensive treatment. But back again to these profiles, so we could in fact see some modifications in the milk fat protein profiles, and especially regarding the small ones. So we could see, for example, that these small milk fat globules were stabilized then instead by these whey protein, milk proteins, quite a lot on caseins, and then we could see also a formation of other bands that we did not manage to identify, but can also be associated with some aggregates. It was a little bit interesting also to see this quite often. These reconstituted milks were composed of aggregated milk fat globules, but as we applied the PEF treatment, in fact, they tended to disaggregate. So there was some differences that the PEF treatment could do in our milk fat globules that we believe can have some effects on bioactivity of those that could be interesting to check. So another study then that we did was regarding the effect of PEF on caseins. And for that purpose, we have used the micelle casein isolate that we have prepared in solution, and we apply to it treatments, to PEF treatments. So first we applied this MCI solution at room temperature for 6 microseconds or 31 microseconds, or we decrease the temperature of the milk to 6 degrees and then apply the same PEF. And basically this was the decreasing of the temperature was to avoid this increase of temperature. So I mean, saying that, we could clearly see the temperature effects that were mentioned before. And then higher temperatures during PEF, in fact, could show also higher modifications of the caseins as observed here. And we could see also increase, in fact, in particle size and yeah, some insignificant changes, in fact, on the zeta size. So sorry, on the for the cold treated samples. So what we were curious was if these modifications of casein would mean an effect on the hydrolysis and then the formation of bioactive peptides for those. So we subjected the different samples, both the cold and the ones that had been run at room temperature, that had larger modifications, and I just showed those here because that's the ones that show high significant differences. So we could see that the fact that these modifications in casein that seem to be small, in fact, could influence the degree of hydrolysis by trypsin. And this difference in hydrolysis, we did a quantification of the bioactive compounds that were formed, and this is just an example of some bioactive compounds that were identified, and it was a number of them, if I remember well. I think was about 10 or 20 that are not founded in untreated milk, but they can be founded in this PEF-treated casein hydrolyzed by trypsin. So I think this show a little bit this potential of producing bioactive peptides by changing the pectin, the casein. We did them in another study also regarding B12 preservation to see if could you preserve B12 by pulsed electric fields. And in fact, we could also see that, in fact, the amount of energy that you apply in PEF does not seem to influence B12. So I think B12 is also well preserved by PEF. I would like now to tell some examples about UVC. And UVC, it is, of course, interesting. I think also Harald mentioned that from an industrial point of view, it's an interesting technique because we have also seen today good examples that microbial inactivation can be reached. And that's also what we would like to check if this can influence these milk components. And I mean, and there is previous studies that have showed that in fact have shown that whey proteins are modified by the application of this UVC light. And you could see that the fraction of the soluble proteins can be depending. This is two different concentrations in this plot of WPI. So in black, and this is 5% solution. But in the case of 1% solution, it decreased. And these differences between two has, of course, to do with how opaque the medium is to transfer of the light, which is also another factor that's important when defining a treatment like this. So we were quite curious then what would happen with sorry, apologize, just saying this. I mean, this was a work done by Casper, so I just put it here because there was also further studies showing this, that the bioactive compounds like lactoferrin and DG and lipoperoxide could be in fact preserved by the use of UVC. So a little bit promising. Can we also, similar what work we did with PEF, can we influence hydrolysis? So that was then the first study that we did. So we used beta-lactoglobulin in this case, and of course has also to do with this to have a solution that is more transparent for the process. And in fact, we could see that beta-lactoglobulin could be modified by the application of UVC. This is also in comparison with heat treatment that we can see here in this plot, as I show. And this means that, I mean, it has been also a modification in the zeta potential. So basically, small differences we could see here in terms of, maybe I should get a laser. So we could see here in terms of our concentration changes with hydrolysis. And also in the total amount of hydrolysates that are formed. But in fact was quite interesting afterwards when identifying the peptides that are formed out of this hydrolysis, that we could really see a significant increase in the amount of peptides that have bioactivity. So it seems to have, there is some potential to create these bioactive peptides by applying these pretreatments. I mean, we even wonder a little bit, and of course this has to be demonstrated, but some of these modifications, we wonder if in fact can affect allergenicity of beta-lactoglobulin, which could be in fact quite interesting. So we continued the work because we thought this was interesting. So in the previous one was just hydrolysis that we have applied. So here we were wondering, what about in the digestion in the human body? I mean, we had also implemented this InfoGest protocol. We have done it with two different conditions, one imitating normal adults and the other imitating the elderly conditions in the stomach, so basically higher pH and a little bit lower concentration of enzymes. And then we could in fact see that the total, and that's what I show you here in this flower plot, the amount of peptides that have some kind of bioactivity is in fact influenced by the presence of this UVC, and of course depends very much on the digestive system, how many of there are formed. I mean, in terms of chemical reactions, of course, maybe for the ones who work more with chemistry are interested, but it seems that there is an increased formation of these thiol bonds, so which mean that this can justify these changes in the hydrolysis afterwards. I mean, this is not our work, but in this context, I would like to put forward this regarding UVC because, as from microbiology, is quite interesting, but it can also be quite interesting regarding formation of vitamin D3. And there are two studies that came up quite recently were showing the potential of applying UVC in milk, and this leads to an increase or formation in situ of vitamin D. And of course, the amount of vitamin that can be formed depends very much on the initial temperature of the milk in the process, but also the dose that is applied. So yeah, further study has been done, quite following, where then it was then applied the processed UVC process at different values to raw milk. And we can see here significant vitamin D compared with raw milk. And after that, we could then see, in fact, that the cream that is produced from this UVC treated milk could also have higher amount of vitamin D. So I think these results are, of course, also interesting. Some messages to take home from my side. I mean, I think there is these technologies have also this potential to modify milk proteins and milk fat global structures. Of course, in some cases, you do not want these modifications, but in some cases, they could also be explored in order to improve the nutritional value and bioactivity of milk. Depends what you use them for and how you explore that. But of course, I also understand that their application has to have tangible advantages. And I mean, and this is very initial work from our side, but I do think that there is some interesting potential in modifying and using these technologies to modify the milk. So I don't have a conflict of interest, I say, but I will clarify that the project has been funded through a PhD project that was supported by Chinese scholarship, and the other part has been a collaboration that I had at Copenhagen University with Arla, and then we had a project Novel Aging that was financed by the Innovation Fund in collaboration with a Brazilian university in São Paulo. And these are the people that have contributed to this project and the results that I have presented here today. Thank you.
Perfect. Thank you very much, Lilia. That's very nice. And again, lots of, I think we're building a very strong and kind of coherent picture of all the complexity of changes, but all the research advances we've made that enable us to hopefully later come back and have a good holistic discussion on where we are now. So I'm going to introduce our final speakers, and in this case, we're going to have two speakers sharing the final presentation, which is on high pressure processing once again, but focused in this case on donor human milk for fat absorption in preterm infants. And we've heard quite a lot on protein, so I think it's great we'll have a presentation that's more focused on fat. And we've two great speakers. We have Deborah O'Connor, excuse me, who's the Earl W. McHenry Professor and Chair of the Department of Nutritional Sciences at the University of Toronto. And we have Dr. Michael Patino, who is the director of the Rogers Hickson Ontario Human Milk Bank in Sinai Health. So hugely involved in both cases in the study of human milk preservation. So happy to hand over to Michael, I think, who's going to kick things off.
Perfect. Thank you for the introduction, Alan, and for the organizers for the invitation to talk today. We'll start with we have no conflicts of interest to disclose. To give everyone an idea of the current state of donor human milk banking, so donor milk is the preferred alternative when there is an insufficient volume of parents' own milk for preterm infants. And donor milk supply in Canada and the US is predominantly by nonprofit milk banks. Nonprofit milk banks like ours here in Toronto, we follow HIMBANA guidelines for safe collection and processing of donor milk. Donors are screened. They are screened by questionnaire, serology. Milk is then collected, pooled, and pasteurized by the Holder method, so 62.5 degrees Celsius for 30 minutes to ensure safety. And pasteurization, as many of the other speakers today have talked about, reduces bacteria to below detectable levels and also can inactivate viruses, including HIV. However, as a result of the extended heating process during Holder pasteurization, and as Casper mentioned, it takes quite a long time for the temperature of the milk to reach the 62.5. There's extended heating, and many heat-sensitive bioactive components are reduced, including immune proteins, but as well bile salt-stimulated lipase, which is an important enzyme, which is important for the digestion of fats and can help with fat absorption in preterm infants. Pasteurization also affects how proteins are digested. We've seen increased breakdown of key bioactive proteins because milk was treated with heat. And while donor milk is great and supports the prevention of necrotizing enterocolitis, which is a serious bowel disease that many preterm infants might be predisposed to, outcomes are often less favorable when feeding with donor milk compared to maternal milk. In these infants. And as alluded to previously, pasteurized donor milk is also associated with reduced fat absorption and growth compared to raw maternal milk. And this is just highlighting that there are several biologically active factors that are impacted by pasteurization, but in particular, bile salt stimulated lipase. So the challenge here is, Holder pasteurization results in loss and denaturation of critical bioactive proteins in donor human milk. So how can we better improve the processing of donor human milk to preserve nutrients and bioactive components to support the recipient infants? And what can we measure to quantify an improvement in processing? So the solution is alternative processing technology. So we've looked at a variety of different alternative processing technologies, including UVC, irradiation, high temperature short time, and high pressure processing, which many of the lectures today have touched on. But for the purposes of this talk and what we've spent a great deal of time interested in is high pressure processing. So high pressure processing involves placing milk into a vessel that is pressurized that then is held for a certain amount of time that inactivates bacteria. So it can reduce pathogens that could be present in milk. Because the process itself can be conducted at 4 degrees Celsius, it minimizes heating and mitigates any losses due to temperature increase. It also maintains covalent bonds, which helps preserve a lot of the nutrients and bioactive components. And to give you a sense, this is the Marianas Trench, which is the deepest part of the Pacific Ocean, which is about 100 megapascals. And the pressure used for food processing is about 5 times that pressure. So it's a significant amount of pressure that's used. So this here is just summarizing one of our early studies where we looked at a variety of different processing technologies, including Holder pasteurization, flash heat, UVC, and high pressure processing. And we found that for both bile salt stimulated lipase and lactoferrin, that we had a significant retention of these components, both bile salt stimulated lipase activity and lactoferrin concentration. But the important part to remember here is that it all depends on the pressure and time that's used. So not all pressure time combinations can lead to better preservation of these components. So in this study, we looked at a variety of different pressure and holding time combinations, as well as Holder pasteurization. And you can see that there's quite a variability in the retention of, in this example, lactoferrin. On the right-hand side, these are 2 separate charts here indicating the retention of lactoferrin post-HPP. And you can see on the x-axis here, we have time, and the y-axis, we have pressure. And there, again, variability on with the retention of lactoferrin and pressure and time for lactoferrin in both lactoferrin and bile salt stimulated lipase. So when looking at studies and when planning studies, it's important to consider what pressure and what time is used. Another thing I'd like to highlight is the use of functional assays. So instead of just looking at individual components, it's often important when assessing new technologies to look at functional assays that can actually measure bioactivity. So in this paper here that was done with one of my former students, cytomegalovirus, which is a common virus found in human milk, was inoculated into raw milk and inoculated into processed milk and incubated prior to measurement. And the goal here was to test the collective activity of antiviral components in milk post-processing. So we used these functional assays to better assess the preserved bioactivity. And in this case, we see that after 30 minutes and 4 hours of incubation, HPP better preserved the antiviral activity compared to Holder pasteurization. So moving onwards, with high pressure processing in human milk. So we also wanted to look at whether high pressure processing would impact the growth of rats. So using an animal model to test whether there is any benefit of HPP or any risk of using HPP. So we looked at HPP in the production of a human milk-based protein concentrate. We noted that many bioactive components were preserved. We saw significant retention of lactoferrin and lysozyme activity post-processing compared to Holder. But importantly here in this study, we were able to demonstrate that high pressure processing was able to preserve the nutritional quality and support normal growth outcomes. So there was no difference in weight gain, food intake, amino acid profiles versus the control. So again, looking beyond just measuring components, but using various models to assess the performance of this new technology. So looking beyond the impact to components in human milk, this was a study to look at how the milk proteins are digested. So as others have alluded to, once milk is processed, it's frozen, and then it's thawed and fed to infants. So we're really interested in what happens once that milk reaches the infant. So we conducted a study where we used a dynamic in vitro digestion model to simulate the preterm infant gut. So we have the stomach, the duodenum, jejunum, and ileum. And we were able to take samples at various points during digestion and measured the proteins and protein activity. So here we were able to show that Holder pasteurization increases the rates of proteolysis of lactoferrin and as well as other proteins like milk fat globule membrane proteins. And here on the right is just a chart indicating that lysozyme activity, post-processing, we see some losses with HPP in this dark gray and Holder in the lighter gray. But you can see that once milk reaches the duodenum, we see significant drops in activity upon digestion. So Holder was reduced from 72 to 7%, whereas HPP was reduced from 75 to 34. So if you just looked at this one here, it tells a very different story than if you looked at the complete picture during digestion. And this is just highlighting that, in the stomach, we have, protein profile, but once we get into the duodenum, that profile is different depending on the processing method. We also, with the same samples, we conducted a proteomics analysis and again saw similar results. And this is comparing raw versus HPP and raw versus Holder pasteurization. And we can see here that while there are some proteins that are depleted in the stomach post HPP, when we look raw versus Holder, we see some, important proteins that are being digested. And these represent the loss of these intact proteins like lactoferrin and bile salt stimulated lipase. So on one end, we have the preservation of bioactivity and nutrients, but on the other hand, we have the, ensuring microbial safety. So it's important given the need for robust validation across various viral pathogens and bacterial strains. So the first thing that we did was we tested HPP and we looked at two different viruses. We looked at cytomegalovirus and hepatitis A virus. These are representative enveloped and non-enveloped viruses. And we could see here that all the different parameters for HPP were able to inactivate them. This is another study, not our study, but a study from another group where they looked at human coronavirus and hepatitis E virus. But the point here really is that there's been very limited assessment of viruses, as well as different processing parameters tested. And this is a common challenge in the field where various groups will look at different pressures and report different results. So it's important moving forward that researchers can come together and work on certain parameters as we look towards new technologies. And finally, looking at bacteria. So there are different approaches to inactivation of bacteria. So you can either look at native bacteria, or you could look at targeted inoculation of individual species. And essentially what we've determined is that HPP is effective. And that 500 megapascals is required for consistent inactivation. As others have pointed to, spores are highly resistant to pressure, and that multi-cycle HPP is an option, but often shows no clear advantage over traditional HPP. But the key here is that bacterial resistance varies by species. So there is different species response to pressure. And we also know from the literature that there is strain to strain resistance to pressure. So there are specific genes in pressure resistant bacterial strains that may express a stress response gene. So it's important, as we work towards understanding the strain to strain variability, it's important to understand how HPP would perform against these various pathogens present in human milk, which again is important for regulatory approval. And then finally, we're working on the development of a surrogate for HPP validation, which is important for determining the total impact of HPP on some of these different baro-resistant strains. So with that, I will pass it on to my colleague, Dr. Debbie O'Connor, and we'll continue the presentation.
Okay, so I'm pulling up the end here. So challenge 3. So while our preclinical data suggests that HPP improves the quality of donor human milk, will these changes result in improved outcomes for very low birth weight infants? And then the other challenge, so challenge B, is how do we begin to assess this? Next slide. So a few years ago, Dr. Patino conducted a scoping review and kind of looked for human studies that looked at different processing procedures on outcomes. And there were no published studies that he found looking at Holder versus high pressure processing in terms of infant outcomes. But there were 3 studies in the literature where human studies that demonstrated that heat treatment impacts fat absorption of very low birth weight infants, and this in turn had an impact on growth. And I'm going to talk about these 3 studies very quickly. Next slide. So this first study was conducted in Sweden by Anderson. It included 5 babies that received either their mother's own milk or their mother's own milk that had been pasteurized using the Holder method. And it was a crossover study. And as you can see here, what they saw was reduced fat absorption and overall about a 17% reduced fat absorption. They did look at growth of the babies. They didn't see statistically significant differences, but with this small sample size, you wouldn't expect it, but it was in the direction you would have anticipated with the reduced fat absorption. In the next study by Stephanie Atkinson, conducted here in Canada, she had 3 groups of 8 infants each. And the groups included a group that received pooled and heat-treated human milk at a 100 degrees C for 5 minutes. The second group were babies that received fresh preterm mother's milk. And the third group were fed infant formula. And I've highlighted the percent absorption on the bottom here. She had the fat balances done twice. And you can see here for the PBM, or the pooled breast milk that had been heated, much lower fat absorption. She also reported reduced weight gain in these babies that received the heat-treated milk. Next slide. And finally, the last study I would like to talk about is a study that included 7 babies. It was a 3-week study where babies were randomized through the study in the 3 groups by random assignment. And babies, they made one large pool, and the pool was either unprocessed milk or was pasteurized Holder or was literally boiled. And you can see in the last column here what the percent fat absorption was. And once again, you can see that heat really impacted the fat absorption, and it looked like the more heat impacted even more. Likewise, this very small study, 7 babies, So too small to kind of do any kind of statistics with the weight gain, etc. But it did follow the trend you would have anticipated with the poor fat absorption. Next slide. So the question for us is we want to see whether this high pressure processing improves infant outcomes. So we wanted to do a study and to look at Holder pasteurization versus high pressure processing. And so we went to the Canadian Institute of Health Research, and we were fortunate enough to get funding to move this along. And this is the study that we affectionately call the HIP study. Next slide. So our principal research question is just that. So you can't go to a granting agency and say, please give me $10 million to do this big study. You have to start with something a little bit more modest to demonstrate that you are seeing efficacy. So since fat seems to be the most studied, and as with the previous speakers, we kind of understand why the fat absorption might be poor, we made this our primary outcome. So our secondary outcomes include in-hospital weight, length, and head circumference gains. And we're also collecting daily morbidities and mortality. And our exploratory questions are, can the intervention actually improve our ability to provide the recommended amount of energy and nutrients? And we will be looking at the blood biochemistries, which are run on babies anyway every week. We will look at residual blood volumes for growth factors, and we'll collect information about cognitive, language, and motor development at 18 to 24 months. The sample size of 100 babies that we're looking at is going to be too small to measure differences, but this will allow us to obtain data in order to build an appropriately powered study later on if we do see positive results with our primary and secondary variables. Next slide. So our inclusion criteria for this study is we need to have the babies in the hospital for 4 weeks because that's how long we're going to feed this. And what we are going to do is mother's own milk is the preferred, and donor milk, of course, is a supplement. So we're going to recruit babies that receive at least 50% of their enteral feedings as supplemental donor milk. So this is going to be double-blinded. We've kind of figured out a way of how we can do this. And the control, of course, is the Holder, and our high-pressure processed donor milk are using these particular parameters that have been shown to work well in our preclinical studies. Next slide. So, and then this is just a schematic. The only additional piece of information from here, again, it's the 4 weeks. We will be doing fat balances after the first 2 weeks and fat balances again after the 4-week intervention. So we're going to do 2 72-hour fat balances, and we'll measure the actual fat contributed from the feeds. We will rinse out all the feeding devices to get the fat that's stuck in the feeding devices, and we also measure the fat in the stools. Next slide. So I'm going to conclude both Dr. Patino and my section. So high pressure processing is non-thermal technology that can better preserve nutrients and bioactive components in milk compared to Holder pasteurization. However, its ability to consistently ensure microbial safety across diverse and resistant bacterial strains must be fully validated before regulatory approval and clinical implementation. And secondly, why high pressure processing appears very strong. Promising clinical studies are required to evaluate whether health outcomes are any better when supplemental donor milk is treated using this methodology versus Holder pasteurization. Next slide. So just want to acknowledge we are in a great position in Toronto that we can do these blinded randomized controlled trials. So just want to acknowledge all the hospitals that participate as well as the research team. And then final acknowledgement, next slide, and just the Roger Hickson Ontario Milk Bank and the wonderful staff and leadership at that milk bank. Thank you very much.
We're good to go. So exactly, if our panelists wouldn't mind turning back on their cameras, and I'm looking forward to discussion to follow. So what I'm going to do is I'm going to take each of our panelists in turn in order of their presentations. and throw together or throw at you some questions, which will be a combination of things which came up during your presentation. Some questions which are asked actually by people while they're registering, and also some questions, obviously, from the Q&A. So we'll go through that. We'll see, and I've got some kind of general questions we might throw up into panel at the end. And obviously, if people think of things, the Q&A is still open. And also, if I can see as we go through it, there's probably a little bit of overlap in that some of the topics or some of the questions others might have a view on. So feel free. I'm looking forward to a good, lively, interactive discussion. So if anybody wants to jump in with a comment, just stick up your hand and I'll call you in if that's okay. So Casper, on that basis, we'll start with you, and I'm going to start with some of the Q&A questions. There's a question about the bioavailability and efficacy, I suppose, biological efficacy of proteins consumed in a denatured form compared to non-denatured proteins after gastric digestion, and whether denaturation, I guess, has either positive or negative impacts on the bioavailability of or the biological functionality in terms of peptides. Do you want to comment on that?
Yes, actually, most whey proteins are more easily digested after denaturation because they unfold, so they become more accessible to proteases. So in that sense, peptides with different functionalities may be formed more easily from these proteins after denaturation. So if we talk about bioactive peptides, denaturation may actually be beneficial, whereas for other proteins like immunoglobulins or lysozyme, which need their three-dimensional folded structure to be active, you lose their functionality. So it depends a bit on the type of component, whether denaturation is beneficial or detrimental to the biofunctionality.
Okay. Now that's really interesting. If I could ask a follow-up. Um, when you talk about denatured whey proteins and its digestion, obviously there's lots of different, we could define lots of different types of denatured whey protein. Does denatured whey protein if we just heat it on its own? Or does denatured whey protein if we heat it in the presence of casein? And there's high pressure processed denatured whey protein. So do you think that applies to denatured? Because I know I'm focusing on it because obviously beta lac is a big topic that several people have spoken about. So the
Mhm.
denaturation and digestion of it's of interest. Do you think all denatured whey protein is the same, or does it depend on the matrix and conditions?
So the denaturation followed by aggregation, and what it aggregates
Hmm.
with does make a big difference. So
Yeah.
if you look at beta-lactoglobulin, its native structure is a very tightly folded three-dimensional structure where, for example, the gastric enzyme pepsin has no access to. As soon as you unfold it, it becomes accessible. But if it then starts aggregating and it forms very tight whey protein aggregates, these actually become again less digestible. So we have seen that if you have a system with only whey proteins, aggregation is detrimental, whereas in the presence of caseins, these whey proteins present on the surface of the casein micelle actually do not show this
Mm.
decline in digestibility as primary whey protein aggregates have. So it's a
Mm.
nice example of how the milk matrix makes this question actually quite complex to answer, and it really depends on
Yeah.
which components are present and how intensive the heating is. So, is it mostly denaturing or also leading to a lot of aggregation? What the answer is here.
Yeah, no, I think it just illustrates very nicely what we've heard in many different ways today, the complexity of milk and each system being different. Actually, and there's another question that you lead me on to, which others might have a view on as well, and again, encouraging everybody to jump in if they have a comment, which is like I always say to students, like one of the biggest, you know, when we heat milk, the guilty agent for an awful lot of what happens is beta-lactoglobulin, and it's basically it's behavior in terms of aggregation interaction. But
Mhm.
human milk obviously doesn't have any beta-lactoglobulin in it. So how does so you would think intuitively the human milk is far more resistant to heat and to heat-induced changes than bovine milk, but that isn't necessarily what we've heard today. Have you
Mm.
or Lily or anybody else got a comment?
Yeah, so some of the human milk proteins like alpha-lactalbumin and lactoferrin show denaturation at a somewhat lower temperature also depending a bit on which study you look at,
Mhm.
compared to their bovine equivalent. So on one hand, the presence of beta-lactoglobulin is leading to unfolding and aggregation of other whey proteins in the bovine system. But it seems that quite a few of the human milk proteins, and then especially also BSSL, which is extremely temperature sensitive, there the protein by itself is already also in the absence of beta-lactoglobulin very temperature sensitive.
Sensitive.
Hmm. Okay, well, thank you. And I'll move on to a question from Jan Trigger Rasmussen. And hi, Jan, for Casper. Do you show the impact of different heating methods on protein functionality and filtration retention? You talked about immunoglobulins, lactoferrin, etc. And were those tested on individual proteins or in mixtures or in milk?
So actually, all the work I've been showing today is really on milk matrix. So
Mhm.
we have not done a lot of studies on heating individual proteins. The only exception is lactoferrin. We have looked at lactoferrin by itself and in a milk matrix. And this is actually a very nice example of what you just mentioned of the beta lac also driving some of this aggregation with other proteins. That
Mhm.
lactoferrin aggregation is way more pronounced in the presence of the milk matrix than without. So uh, yeah,
Yeah.
it's a nice but complicated protein in that sense.
Absolutely. And we have another question in the Q&A from Bruce German, and hi to Bruce. And he raises a point actually that I think a number of people might have a view on, and Harold, you might have a comment on this as well, that that if we're talking in the future of processing being combinatorial about involving different mechanisms like combining heating and filtering and UV or whatever it might be, but it's the challenge to taking what might be it's the scale challenge between bovine and human milk. Where obviously in bovine milk we're quite used to unit operations with big pieces of equipment, and the scale of the challenge of applying this in the future. And as Bruce puts it, what do you think it'll take to recruit a generation of engineers to multiplex and scale the unit operations of milk down to milk bank level? And I was also thinking this, Debbie, during your presentation about where do we see this, despite all the advantages of HPP we're hearing for human milk, where will it be applied? And are any human milk banks in a position of buying a HPP unit, for example? So I don't know, Casper, do you want to take it? Then I might hand over to anybody else who'd like to come in on that one, because I think there could be quite a few different perspectives on it.
Yeah, in the end, I mean, we are now looking at, together with the Human Milk Bank in the Netherlands, at how can we downscale some technologies. But for sure, it will not just be a miniaturized version of something you would see in a dairy factory. And also,
Yeah.
other technologies may be more or less feasible in a human milk bank. I mean, the high pressure that was presented by Michael and Deborah in the final presentation is a nice example. I mean, this is something that is feasible within a human milk bank setting, but I don't see this in any way being implemented in industrial dairy processing. But maybe Harald would like to follow up on that.
Yes, yeah. So if I look to high pressure processing and full scale, so if you look to dairy processing, that is also always continuous processing. So from a high pressure perspective, I know there are big scale machines available, all for impact pasteurization. However, this would never be manageable within a dairy factory. So then you would really have a specialized product, and then the high pressure unit would be single for this product and really low throughput. But for mainstream products, I don't see that. But other technologies work a bit the other way. Yeah, so some technologies can be scalable to big size, and also back to human milk banks to small sizes. But it is really an engineering challenge.
But just Harold, as a follow-up, is it not correct? I'm not sure how far it went, but that there was interest in commercializing for use in colostrum processing in New Zealand. Wasn't there a commercialization of bovine colostrum, as it happens, using HPP in a commercial process? And I always think for HPP, it's going to have to be a very high value, low volume
Yes.
product to work and to justify the cost and the investment. So do you think there are niches in biological fractions that might come out of milk. I mean, we're increasingly pulling apart milk into a million different directions for value. You know, a very low volume, very high value, where there could be a niche for high pressure processing.
Yes, for sure. And that's actually a bit what I meant. So for mainstream
Mm.
processing, I don't see a future within dairy processing. For instance, for
Yeah,
fruit
for
juices,
bulk
there's
processing.
quite some mainstream HPP processing because it's just
Mm.
somehow practical and there's a quite high value-added product in that sense. So you can easily make the costs for the high pressure. Mainstream dairy, so raw materials are almost as expensive as the product costs in the end. So
Yeah.
so commercially, so there's not much room to invest in high pressure equipment, both from a capacity point of view, but also from an investment point of view. But indeed, if you really have high value added product where where HPP can be the solution, then I see possibilities. I also have seen several publications or reports on HPP, indeed on colostrum, but also on just on milk,
Hmm.
as a separate, just on fresh milk instead of pasteurization. From my perspective, for Friesel Campina perspective, that will not work. But if you are in a niche market where you have a really high value, then it could be
Yeah.
beneficial.
Okay.
Yeah.
And just on the milk bank issue and the practicalities of HPP, despite the I think accumulating evidences we've heard of the benefit. Michael or Debbie, do you want to comment on the practicalities?
Well, I can start. And
Mhm.
so there's over 700 milk banks, human milk banks in the world. If it's a milk bank just for one hospital, I don't see that it's very practical. But something like the Roger Hickson Ontario Human Milk Bank that supplies the entire province of Ontario as well as the Atlantic provinces,
Mhm.
appropriately scaled piece of equipment is extremely feasible. And you have a lot of labor costs, right? With Holder pasteurization in terms of the time, your throughput could be much quicker with an appropriate scaled machine. All that being said, probably would not incorporate it unless we see improvements in outcomes.
Yeah, of course. Okay, Michael, do you want to comment on that?
Yeah, I mean, I think Debbie touched on all of it there. I mean, from a milk bank perspective, we're always looking on ways to make, a better product and make it more efficiently. So high pressure would definitely be one of the answers to that, one of the solutions to that problem. But again, it does have significant startup costs. So I mean, if
Mhm.
you're in a position to be able to purchase such a machine, I think it would be valuable. But again, from a global milk banking perspective, I think we also need to look at other technologies that might be able that we can use to implement in other milk bank settings that can't necessarily where they can't necessarily afford to purchase such a high ticket item. Yeah.
Alan, you're on mute.
I was trying to save everyone the noise of my barking dog in the background. So apologies. I thought I'd come back off mute. But yeah, and it's interesting how it might compare. I understand there are some people looking at spray drying as a very basic way of preserving and storing human milk, but that raises a whole other set of perhaps issues about reconstitution later. Dave, we might move on to you if that's okay. Um, and you talked about antimicrobial defenses. I think an interesting thing to think about is that both milk and both bovine and human milk have a natural range of defenses, whether it be enzymes or proteins like lactoferrin. And how do we like how powerful are those and how much are we damaging the antimicrobial defenses by processing?
Mhm. Yeah, I mean, for sure, the specific antimicrobial proteins, a lot of them are getting degraded by Holder pasteurization or much more by retort processing or ultra high temperature processing and being preserved. Um, But yeah, then the question is, how much does that affect, antimicrobial activity within the baby? And how much does it, impact risk of infection? That part we don't really know very well. I mean, Mike made the point about lysozyme preservation that was higher with HPP, which was a really exciting finding. Go, Mike. Um, I think that kind of work needs to happen more and to examine like the that survival. Um, we have done some preliminary work in our lab to show that there's higher levels of lactoferrin preservation in the stool of babies after high pressure processing versus Holder pasteurization. So that could be another indicator. But yeah, the work still needs to be
Yeah.
done.
Okay, and Dave, you mentioned something in your presentation that I know is of interest to both of us, which is proteases in the milk and the
Mhm.
inactivation of elastase, for example. But do you think there's evidence that some of those proteases have important biological function of their own in terms of assisting digestion or producing peptides, and again that inactivating them mightn't necessarily be a benefit?
Mhm.
Just curious on your thoughts on that one.
Well, yeah, so that's still very much an unknown, I would say, right? I mean, like similarly to, our concern about lipase and,
Hmm.
that's based around the fact that preterm infants have low endogenous lipase production, and that human milk could be beneficial. Preterm infants also have low protease production in both their
Hmm.
stomach and their intestine. And so the concept is that the proteases in the milk may be helpful, like they are for lipase. But we don't have a strong evidence yet about the benefit of proteases in human milk.
Yeah.
So it's an idea, it's a theory, but it still hasn't been tested very clearly. I think I found one paper that was indicating some potentially higher protein absorption with raw milk versus Holder pasteurized milk. But again, I think it still needs to be examined.
Okay. Okay, thank you. And going back to the chat, there's a question here, and I'm not sure if you or anybody else, it's a fascinating question, but I don't know who might have the answer to it, which is there any information on how climate change is affecting lactation and lactating women? I'm not sure who wants to take that one, if anybody has any comment or has any awareness of research in that. I think it's a question I'm sure we're going to have to face at some point, but I'm not sure if there's an easy answer. We might move on. There's an interesting one here from a Lucas Wiza. Are there unwanted bioactivities in milk? Are there things in milk that are not microbiological that we want that we could view as hazards?
Well, from like a protein or lipids, anything that's synthesized by the
Hmm.
mammary gland or absorbed, those things pretty much we consider everything as beneficial in human milk. Um,
Hmm.
there are, beyond that, human milk can contain various environmental toxicants that can
Hmm.
be, if that are consumed in the diet.
Hmm.
Usually the levels of those are not like at a concerning level. They're not at a level that's, problematic, but they
Hmm.
do exist. Yeah.
Okay. And we have a question here from Betsy Church for you, Dave, about why you chose bile salt simulated lipase as opposed to LPL, and what process did you use, what assay, I guess, did you use to measure it?
Yeah. So, and this can be for Mike and Debbie as well, since they're working with
Hmm.
BSSL. But, BSSL is a more important lipase when it comes to overall fat digestion in infants than lipoprotein lipase. It just has more activity during digestion than lipoprotein lipase does. And so that's why we focus on that protease.
Okay.
Mike and Debbie, do you have anything else to add?
Yeah, I mean, I think from papers that I've seen, about 90% of the lipase in human milk is BSSL. So I think just from that perspective, it makes more sense to look at BSSL.
Okay. Okay, thank you. And we have another question for Dave, although I think it also um others might, Lily and others might have a comment on it as well. It was from again from Jan. What's the impression of damage caused by heating on bioactivity of the milk fat globule membrane components, proteins and lipids?
Um, yeah, so there's definitely damage to a variety of different milk fat globule membrane proteins with thermal processing. That's for sure. They are, pretty sensitive to thermal processing. I mean, especially, I mean, they're definitely more sensitive than casein proteins, for example.
Mhm.
But Lilia, do you have any other thoughts on that? You're on mute, by the way.
Yeah, I open it. No, in fact, we were also quite curious about that. I think it was quite interesting to see that they could be released from the milk fat global membrane, and they are quite small, in structure. So sometimes means that they could also be better preserved, but we don't know enough. I do think this is an area that, in fact, is interesting to investigate further. So, but I have no idea, in fact.
Okay.
I think
Mhm.
Casper has done some studies related to this as well.
Casper, do you want to jump in there?
Yeah, but the answer is not that easy here.
No.
I mean,
You have no easier answers to that.
no, no, sorry. I'm maybe that's just to make sure that I remain busy the rest of my life, I guess.
And another question, which again could go to Dave or possibly Debbie and Michael might jump into, and it's from Bruce again. And he says, wonderful studies on preterm infants and the implications of BSSL to fat digestion. Do you see much variation in BSSL levels among different milk samples, depending on stage of lactation, etc., with the idea that maybe donor milk should be assayed if targeting preemies? Do you think it's a fairly consistent activity or do you think it's fairly variable?
Yeah, that's a great question. I haven't done the study. I think in literature it might vary like up to like twofold or so between people. So yeah, that variation could definitely affect the effectiveness in the baby. There's probably also changes that occur across time. A lot of milk components change across
Yeah.
time.
Hmm.
So, yeah, I mean, it's possible that that is something that we would want to measure in milk and,
Yeah.
Mhm.
perhaps optimize, if we were talking about donor milk, which is usually mature milk from later in lactation compared with the earlier earlier milk from a preterm lactating parent. They could have different levels, and maybe that donor milk needs to be slightly optimized further. And I don't know. There could be different routes for optimizing, with maybe pooling practices or even possible like fortification with recombinant proteins
Hmm.
is another possible direction.
Mhm.
Um, and then, uh, what was the other part was about the lipid size, is that right? Or
Yeah, exactly. Do variations in fat globule size and composition affect digestion in actual infants?
yeah, well, I don't know, but I think that smaller milk fat globules may be more rapidly hydrolyzed than larger ones just because they have higher surface
Yeah.
area.
Hmm.
Um, so it's also possible that if the ranges of sizes are different between different milks, then maybe there's different rates of digestion.
Mhm.
Maybe, I don't know, Lilia, you've studied these sizes, so maybe you can add more to that.
Yeah, I mean, now, to be honest, I do not remember all these details, but I do think that the smaller ones, I think it has been a study where they compare the effects of these small milk fat globals versus the bigger fat globals. And if I do remember well, I think they also thought the small ones are more healthy or could have better health benefits. But now I do not remember the study with the detail. So, and of course, can also depend very much how much protein they contain, right? In the because I mean the membranes in a small one is a larger part of the global,
Yeah.
right? So can also be that you get a more dense, feed or material. Mm.
I did want to add just an aside here. So related to that, in our study that I was mentioning, it's not published yet, but with the fat absorption from the HPP milk, we did find that the absorption was higher for like shorter chain, medium chain fatty acids than the longer chain fatty acids. In those samples, like the difference was it was more improved. The fat absorption was higher, was better with HPP compared with HOP for those ones. So our thinking is that perhaps it's not just a limitation in lipase activity, but perhaps also like a limitation in bile salts that are present
Correct.
in the gut. And so there's still, some issues with the absorption of those longer chain fatty acids, which may require more bile salts for absorption.
Mhm.
And maybe one
Makes
more
sense.
question for now on the human milk, and I'll move on to Harold and Lilia. But one thing I was very interested to hear from a number of presentations, and this question might come from my own ignorance as opposed to anything else, but is the number of references to viruses. Because I always
Mm.
have this kind of intuitive feeling the viruses are kind of unknown territory when it comes to milk compared to bacteria. They're much easier, bacteria are much easier to
Sure.
measure and study, etc.
Mhm.
Like, do we have a big knowledge gap regarding, do we have blind spots regarding viruses in milk, whether it be bovine or human? And how are we doing in research in that field? As I say, I just haven't tracked it compared to microbiology, conventional bacterial inactivation. Does anybody want to jump in with viruses? Is that a big area should be doing more on?
Now, it's indeed a very complicated one. With human milk, it's especially CMV, I think. Yeah, sorry, Lilia, for my. But for human
No,
milk, it's
but
especially CMV that is an issue, and we have been doing some tests with CMV inactivation, but also how well it can still infect cells. But if you compare this to our pathogen testing,
mm,
I mean, pathogen testing you can quite easily and quickly do if you have just a safe lab. But these virus testing, and especially the infectious capacity of a virus, it's way more complicated. So I think that's
mm,
also one of the reasons that not a lot of people uh, actively study viruses. But it is indeed a concern.
yeah, I'm not
Anybody else?
aware either of many studies in this area, in fact.
Well, Mike Patino wrote a whole systematic review about this, so you should chime in.
Well,
Sounds
well,
like you've just lined Mike up for
Well,
I'll sit
no
back.
pressure, Mike.
well, I'll say that in the human milk literature, I'm not too familiar with the bovine milk literature and viruses, but there have been, I would say, probably 10 or so studies that have looked at a variety of viruses, mostly CMV. Some have looked at, your Ebola, Marburg. We actually published a paper during the COVID pandemic where we actually looked at inactivation of COVID. The SARS-CoV-2 virus in human milk with Holder pasteurization. But again, very limited for alternative processing technologies. I mean, there are many
Mm,
viruses out there, and we just don't know
mm,
how they might impact, and how they might, be inactivated, both in terms of their structure being, destroyed and their infectivity.
mm, mm.
Okay, well, I'm sure we'll come back to human milk, but I might move on at this point to Harold, and that was a really interesting. By the way, this feels like the strangest. I feel like I'm conducting the strangest high-level PhD viva I can imagine. It's like with all these questions. But Harold, we talked about HPP and scale. That was one of the things I wanted to talk to you about. But I'm curious as to how you kind of came down quite well on the idea of UVC. But
Yeah.
what do you see are the barriers to UVC adaptation? I mean, we heard about the problems, the opacity of
Yes.
milk, the need for high surface areas, but do you see that that's a scalable, applicable technology compared to?
Yeah, that's a very good question. So we do a lot of internal research also towards UVC, and what we see. So to make it scalable, you need to use something else, because UVC is not, or milk is not UVC transparent, or dairy streams. So you need to create somehow turbulence in your system to make sure that every particle of your milk, every liquid particle, will be treated. What we have seen in the recent years, several companies pop up that produce UVC equipment on industrial scale, which can really go up to scales which are suitable for industrial production. So, for instance, 20,000 liters an hour or 30,000 liters an hour. So in that sense, from scalability, I see a large opportunity for that part. However, what is lacking is really a good view on legislation and adaptation of the technology. So on the one hand, in Europe there are some either non-novel or novel food agreements on different types of dairy products already. In the US, some things are happening, but if you look to a global scale, it is really difficult to interpret if they would accept, from a legal perspective, if UVC is a good solution. Another part is also what was explained by Leah, is that we have this vitamin D increase in dairy products. That seems nice. From one perspective, but from the other perspective, if you have strict vitamin D regulations on your product specifications, that can implement problems.
Yeah.
So especially if you go towards infant formulas, you have to take that into account, that you have this vitamin D increase in your products.
And that's very interesting. That too much, you could go too far in what would appear intuitively to be a good direction. And Harold, you partly answered my next question, which is actually going to be about legislation and about in the EU. I mean, I think many of us will be well aware how long it took Danone to get approved for the first HPP product, which was strawberry pieces back in the 90s. And it took
Yes.
years and years. And HPP went on a long, slow, multi-decade journey to being no longer regarded as a novel technology. But where do the rest of the technologies fit? Like, is PEF currently allowed for use as a technology in Europe? I guess UV is probably, but and how much are barriers that, despite the science,
Yeah.
accumulation of
I
evidence?
want to put a different, it a little bit in a different perspective. So, like I said, for some technologies, it's unclear if you may use it, just as a technology to produce your product. But on the other hand, you have another discussion, and that is more if you want to use a new technology as a substitute for say what we call the legal pasteurization within milk production.
Right.
So that are two different levels of looking towards it. So if you really want to say, okay, my new technology, whatever it is substituting the only microbial barrier that we have within the production, the only microbial reduction step, like normally is the pasteurizer, which is legally allowed, and also in a lot of countries for a lot of products obliged by law. If you are going to change that is much more difficult than what in several presentations was demonstrated, is that if you use just legal, let's call it legal pasteurization, and have additional technologies next to it. Because then the question is not, does the technology reduce enough microbiology? Because that is the responsibility of the food producer to produce within specifications. You covered, say, your food safety part with heat pasteurization, but you still need to comply to your specifications as you sell it. And so that is a different discussion. And there the discussion is only, can I use it? Is it allowed to use it as a substitute, as an extra technology in my supply chain? Which is a bit easier than if you are going to say, I'm going to replace my pathogen removal step, which is now the heat pasteurization step. And Yeah, what we see is, despite some lacks in legislation, but if you look at it a little bit broader, then, for instance, pulse electric field, but also high pressure is now used quite often. But pulse electric field less for microbial reduction. But I sometimes ask also internally, how do we look then to microbial filtration technologies?
Mhm.
But there the discussion is already different, because microbial filtration technologies do not do anything to the components in your product. You
Yeah,
might remove them, or they
they
go
might
through,
change them. Yeah,
but there is no chemical change in that process. So
yeah,
there's quite some things
yeah.
to think about.
A
And
bit
the
like
more,
bacteriophagation, which you mentioned as well. Yeah.
yeah,
Mm.
yeah.
The
And
purely
I
physical
think
process.
the more potential reactions you would expect, the more difficult it becomes.
Okay.
Mhm.
Yeah.
And just one more question. And you touched on it again there, preempting, which is about consumer response. Another, if
Mhm.
you like, barrier is going to be consumer response. And I remember my group, the first paper on 3D printing of cheese, and we were very excited about the potential of 3D printing. And then, I had a PhD student do a consumer study on 3D printing acceptance, and people were like, "What the hell are you doing trying to 3D print food? That sounds really
Yes.
weird." So there
Yeah.
was a gap between the scientist's enthusiasm and the
Hmm.
consumer's reaction. And in that line, like Bruce asked an interesting question. From your perspective, what biological values do you see being appreciated by consumers in the future
Mhm.
to drive innovations? So what's a big enough value or benefit the consumers would pay enough for the
Yeah.
premium you might add to something
Yeah.
like HPP?
Yeah. That's a good question. First of all, I really agree on that you need to promote the benefit,
Hmm.
and not the technology. And by the benefit, you can maybe explain the technology, yeah, why you use it.
Mhm.
But if I look to commercial dairy production, where the most valuable part is within for non-thermal technologies, whatever it is, then is really in producing the infant formula and going as much as possible towards human milk. So the golden grail, the holy grail, sorry, is to get as close to human milk as possible. So if there are components that are heat sensitive, but which are to be known to be similar as in human milk and beneficial, then it's worthwhile to keep them.
Okay.
That you can use in your story about your infant formula.
Mm.
So that is one. That is the main part. For if I see to more the adult nutrition part, so the normal consumer products, then you go more to, for instance, sport nutrition or elderly nutrition, where you
Mhm.
know from all the research from which
Mhm.
components can be beneficial for what reason, for building up muscles,
Mhm.
or keeping muscles in elderly people, all these kind of things. And if there are components related to heat sensitive, then these technologies come into place.
Mhm. And there's an interesting question there. Maybe we're getting back into the human milk discussion a bit, but somebody mentioned a couple of minutes ago about recombinant proteins. But it might be an interesting
Mhm.
question to whether we'll see in the future products being treated by heat, particularly if you think about human milk or infant formula type products, where we're getting back, an activation, we're getting the microbial safety, but then we're adding back in the biological functionality from another route, like recombinant or something. I don't know if that's a to
Yeah,
put back in
that
your
can be part of the
lactoferrin.
that can be part of the strategy. Yeah. So depending on how you make your infant formula, you can
Hmm.
mix in the powders. You can
Yeah,
mix
dry
in
blend
certain
in. Yeah,
ingredients, which have these benefits,
yeah.
and that
Okay,
can
well,
also maybe also come from liquids, but normally it comes from the powders.
Mhm.
okay. So maybe we'll move on, Lilia. And we've had quite a bit of discussion about PEF. I remember I was at a conference in Germany a number of years ago, and there was a company called Elia, I think, who were
Exactly.
selling,
It's
and they
still
had a
there.
machine. They had a machine called Cool Dairy, which they were describing as a milk PEF pasteurizer.
Mhm.
I just wondering, where do you think PEF really is at somebody else told me they saw the potential, interestingly, as really being in the lower volume, high value, like protein isolates. trying to retain functionality. Do you think there's a realistic in 10 years, are we going to see PEF being used more widely? Because the equipment seems to be there.
I mean, the path is there, right? And it's, I mean, right now the most or the most common application is used for softening of potatoes, right? So maybe
Yes,
you are
yeah, for
aware
chips. Exactly. Yeah.
because that's where it found its place to the market. I mean, regarding with inactivation of microorganisms, there are some challenges that are difficult to overcome because you have to combine also with heat treatment
Mhm.
probably, which then loses the advantages. I mean, we start to um, consider these possibilities, like to modify, if you modify the proteins in some way that affects the hydrolysis, and then you can
Mhm.
produce more bioactive compounds that could be identified, and then eventually afterwards um, extracted or separated,
Mhm.
and so you could get enrichment in those. Then maybe there is a potential of using PEF for modification of proteins. So otherwise, I do not think. I think they have been trying for quite a long time. I think
Mhm.
there is what has been said, and I'm sure Harald knows better than me. But I also think it has to do with volumes, and then
Yeah.
even niche products. It's difficult to get these tangible advantages that you will pay for the technology. That's
Okay.
my belief.
Okay,
Yeah,
that's very helpful. And
Harald, maybe you can.
if I may add,
Yeah. Mhm.
for microbial reduction with PEF, acid environments are really beneficial. So and
Correct.
dairy is normally neutral, so
Yeah.
that makes it really hard to prove
Hmm.
sufficient or wanted amounts of reduction of microbiology. So therefore, it's
Yeah.
besides the scalability issues, you also need
Yeah.
really a lot of
Yeah.
relatively a lot of energy to make sure that you reduce
And
sufficient.
the microorganisms that get injured, if you want to say, I do think they recover in such a
Yeah.
nutrient-rich
mm,
environment. That's also probably part of the problem.
and that's an interesting point actually, Lily, because there's a question in the Q&A in the similar theme, but it was talking about, I think, on proteins and MFGM proteins, where some of the effects you saw reversible over time, or are they irreversible?
I mean, these perforations of the cell membranes are irreversible, but of course depends what kind of the intensity of the electric field that you apply and what
Mhm.
kind of process you apply afterwards. For example, if you shear a lot of the things after you apply the electric fields, then you for sure destroy all the membrane because it's a question that if the membrane is unstable, and then you
Mhm.
don't do anything, then it can recover. But if you apply also, I mean, pumping, I'm giving you as an example, is more likely that you destroy, that it cannot be reversible. And especially in milk, that, for example, then caseins can get in the middle
Mhm.
and stabilize because they're also good. Then it's very difficult for the B layers to recover their original structure. That's, I think, what
Okay.
we have seen it.
And thanks, Lillian.
Mhm.
I'm just actually keep conscious of time, so I'll move on. And there's a question moving on to Michael and Debbie again, and hopefully just round up with a few final questions. And Bruce again in the Q&A said, inspiring as always. And I wonder why in Toronto you call the study hip check. The milk fat
Yeah,
globule,
exactly.
the milk fat
I
globule
was
membrane.
also wondering. Mhm.
Uh, and Bruce is wondering if there's variation in the ability of preemies to absorb complex lipids versus simple triglycerides, and whether that was going to be part of your study.
Well, maybe I can start. So
Hmm.
we are going to recruit 100 babies, so we'll have a reasonable sample size. All the babies will need to have be in the unit about 4 weeks. So we transfer out pretty fast in level 3. So these will be pretty small babies, anywhere from about 23 weeks post-conceptual age till probably about 32 weeks. So we probably will have enough sample size that we can look at how old the babies are in terms of whether that has any influence in the two groups in terms of fat absorption. I mean, listening to this conversation today, I think I sort of had in my mind that we were just going to look at total fat in the feeds and in the stools. But what I'm hearing is we might want to also look at the types of fatty acids, at least from based on your work, Dave, for sure. So there might be other things that we should be looking at. I think where we are just in terms of the regulatory piece in Canada, donor milk's considered a food. So in fact, we do, and high pressure processing no longer has novel food status. So there's no pre-market assessment, means that a clinical trial is not required to do this. However, we're working really closely with Health Canada to make sure that they are satisfied that in the context of everything else that's going on in the milk bank with screening, etc., that it can be as safe as donor human milk. All that being said, it gets back to the point, it doesn't make sense to incorporate this technology if it doesn't yield impacts in terms of clinical outcomes. So I'm not sure if that answers your question, Bruce, but it maybe kind of gives you something where we're thinking, how we're thinking of doing things.
And Mike, in your presentation, and thanks, Debbie, in your presentation, you mentioned something that caught my interest. A lot of things caught my interest, but I'll just pick on one or two of them. One of them was human milk protein concentrate. What is that? I mean, we're familiar with bovine NPCs and fractions, but I never heard of a human milk protein concentrate. What's the idea of that?
Yeah, so this was a project a few years ago, maybe more than a few years ago now. But the goal was to use some of the milk that we can't use for processing, because it might be higher in bacteria that we would typically discard. And we thought, how can we add value to that, and how can we recoup the value and the investment in that milk? So we had worked on developing a human milk protein concentrate through a variety of different concentrating steps with microfiltration and spray drying, and then the final step was the high pressure processing to inactivate the bacteria that were also concentrated as you concentrate the protein. But it's something that we worked on, and we're hoping to continue working on, because as the director of the milk bank here, we're always looking for ways to reduce waste, especially when milk that we receive, we don't have a field of cows that we're milking. So this represents actual donations from humans that are spending lots of time. So just in terms of our sustainability is something that we're working towards.
And actually, there was a question that was raised, I think, by somebody at point of registration, which struck me, and I'd be interested. Like, one of the simplest technologies, not for bovine milk, but for human milk, and possibly commonly applied, but hasn't really been mentioned, is freezing, which we know that mothers can do for various reasons. And freezing possibly combined with things like microwave thawing and different thawing practices. And there's a question, and it was in my mind as well. Like in fact, my first work that I ever did in human milk was a question about fat destabilization on freezing. And we did some work on that. And I'm just curious to know, what about freezing in human milk, and what impact it might have on biological factors, or how much do we know? Because domestically it probably happens.
Yeah, I mean, we reviewed the literature on this as part of some work that we were doing for the WHO, and looking at different freezing practices and the impact on bioactive components in milk. And there seems to be some evidence to suggest that freezing does impact some proteins.
Hmm.
But again, the variability in freezing techniques, whether it's in a fridge freezer, if it's in a deep freezer at minus 20 Celsius or at minus 80, it's sometimes, we weren't able to do any meta-analysis on any of those studies because there was just too much variability and too little studies that were actually looking at the same thing. So
Okay.
definitely, I think from a practical perspective, looking at freezing is important. From a milk bank perspective, we do provide some recommendations to
Mm.
donors who are sending in their milk on how to safely store their milk. But again, a lot of that is out of our control. But I think again important, especially for reaching a larger audience and
Okay.
parents out there who are doing this every day.
And I think in the interest of time, I'm sure Jenny's going to probably come on shortly to wrap things up. So I'm going to maybe ask two final kind of global questions that anybody might take. And one is from the pre um questions we got. And I think it's really interesting. I think it probably applies more to human, but maybe it applies to bovine too. How do we prioritize, or I guess balance, improving safety at source in terms of collection and testing rather than post-process remediation? How much is how do we weigh up improving, standards of raw material, if you like, as opposed to trying to fix it with a process? And I'm just curious as anybody, I thought it was an interesting question. Does anybody want to throw in something on that? I guess it's something we can't control is the raw material, whereas we can control.
I think that's a super, super important question. I mean,
Mhm.
obviously, if we can prevent contamination at the source, that's a really good thing. I think, and so, in some cases, pasteurization is uh, possibly overkill for some
Mhm.
samples, right? Like, for example, like in human milk, often like there may not be a very large amount of bacteria or even pathogenic bacteria in any particular sample. But we
Mhm.
still do Holder pasteurization as like a just in case measure, right?
Yeah.
Because some samples may have something.
Yeah.
So, yeah, if there was a way to ensure safety without processing, that would be great. Definitely, but it's challenging for sure.
Yeah.
But I know that newer like testing methods for, testing the bacterial, composition of raw milk are improving, for more rapid testing. So maybe that's a direction that could go in the future.
Mhm.
And then the other thing is like maybe we can add other, biological compounds to milk to help
Mhm.
prevent microbial growth without these more intensive treatments.
Mhm.
So,
Yeah, I remember some studies on adding an enzyme called lactose oxidase,
mhm.
which was able to generate at a food grade level antimicrobial compounds. But look, and I see Jennifer has, so I'm going to finish. And I maybe this won't be a question, but I just make one final comment before I thank everybody, and that's and we won't have time to discuss it. But Mike, you raised another one which I thought was very interesting. Because you referred to surrogates, and something that made me think about is where's the alkaline phosphatase test for these processes, whether it be for bovine or human? Where is the validation that we know that it has worked without having to go through? And where is our potential? And do we even use alkaline phosphatase for human milk, for example? It's an interesting question. But how do we know these have worked without going through? And where are we on the surrogates? But I think we'll have to leave that one as a cliffhanger because we won't have time.
Oh,
And
come
I'm going to
on.
thank
No, there's 10 minutes
anybody.
for
Okay,
that.
I've tweaked. Anybody want to jump on that one?
I'll jump on it very, very quickly. So we are working on developing a bacterial surrogate that we can use here at the milk bank because when you're working with these different bacterial pathogens, you can't necessarily use them easily clinically. So we're working on identifying the most barrel resistant strain of bacteria that we could find and then cross-referencing that baroresistance with some non-pathogenic bacteria that we can easily use and incorporate into each processing run, so that we can measure that as our indicator of pasteurization. At the milk bank here, we test all of the milk post-pasteurization anyways for bacteria, but we don't necessarily have a enzymatic test like your traditional alkaline phosphatase for bovine milk.
Yeah.
And it wouldn't work because alkaline phosphatase in human milk is more temperature sensitive than that in bovine milk. So they don't provide the same type of
Indications.
data.
Yeah.
It's also
Well, that's good.
Yeah.
It's also preserved by some of the non-thermal technologies, so you wouldn't
Yeah,
even
yeah.
see it.
HPP isn't very
So
good
if you
against
look to
alkaline phosphatase, I think.
if you look at bovine milk production, this is also a challenge. If you would replace
Yeah.
heat pasteurization, and then you really need to have it built into your process that you can make sure that because it's only an indicator. If I look from an industrial perspective, an indicator saying that I have sufficient time and temperature. So you need a different kind of indicator that you have, for instance, enough pressure and time.
Yeah.
If you relate it to pressure, something that is sensitive to that to be measured.
Okay, well, I think we'll call it at that point. Thank you, everybody, for I think a fascinating, and I hope I've covered most of the questions that were submitted on the Q&A or in advance. So thanks everybody for your inputs. I think it was a great way to wrap up with that discussion. I really enjoyed our very strange kind of PhD Viva-like experience for me here, and I'm happy to hand back to Jenny, and thanks to the IMGC for inviting me to moderate this evening or this morning, as the case may be. I really enjoyed it. Thank you.
Thank you so much. That was such a riveting discussion on the very many challenges, it seems, that our field is facing. But there's also progress, immense progress in the tools and discoveries driving forward. So I'd like to immensely thank our brilliant speakers and our moderator for taking their time to share their invaluable experience and insights on the topic. Now, the recording of this webinar will be available on the IMGC website on June 15th, not July 1st. And if you've subscribed to receiving notifications from IMGC, we'll let you know as soon as it's up on our website. So the topic for each year's Roll Up Your Sleeves Ross workshop is proposed in part by attendees. So that would be you. So from the previous workshop. So please fill out that exit survey. And then the topic is fine-tuned by, and speakers are identified from within the IMGC Scientific Advisory Committee. So we have a subcommittee, and this year's subcommittee, which I would like to make a big round of applause for, is Alan Kelly, who was our moderator today, David Dallas, and Casper Hedinga. Okay, so as a nonprofit organization, IMGC's activities are, supported by by funding from sponsors, our partners. And I'd love to thank our elite sponsors this year. So that's Our Love Foods Ingredients, By Heart, a National Dairy Council, and California Dairy Research Foundation, which also co-manages IMGC. If you're interested to also become a partner, please contact me and I'll be happy to chat with you about what that looks like. Okay, so finally, scientific advancements cannot be made without the brilliant and passionate scientists and innovators. That is our base, and that includes all of you. We rely on your partnership in advancing the field, and our success really depends on your insight and your input and really your engagement. And so there are 5 ways that you can get involved with IMGC. So if you haven't already, please subscribe to Splash Milk Science Update. This is our bi-monthly publication on milk science for human health, and share the articles with your network. You can also join our LinkedIn page to network with fellow milk scientists, milk enthusiasts from other fields, and you can learn about job funding, internship opportunities, and all the future events that we host. Next, please complete the 5-minute exit survey for today's workshop. This is so important because your voice really matters. It contributes to developing next year's workshop or designing it. And then another way you can engage is to join various working groups and co-author review papers on specific milk science topics. So if you're interested, just let us know in that exit survey. And finally, I hope to see you at this year's 23rd annual IMGC Symposium. It's co-hosted with Oregon State University and Oregon Health and Science University in Portland, Oregon on September 15th through the 17th of 2026. So that's this year. We are still accepting poster abstracts to host during the poster reception. And so you can find more information about this event on our website. Thank you for participating here, and we will see you all soon. Bye.